There was a time when nothing was trying to be alive. Not empty in the way we imagine emptiness today, not a quiet room or a dark sky, but a deeper absence, a universe with no preference for survival, no awareness of continuation, no concept of before or after. Matter existed, energy moved, forces pulled and pushed, but nothing cared whether it lasted. Nothing remembered. Nothing hoped. Nothing feared an ending. And yet here you are. You are made of atoms that once belonged to stars, arranged into molecules that should have remained indifferent forever. Somewhere along the line, something impossible happened. Matter crossed a line it was never supposed to cross. It began to resist decay. It began to copy itself. It began to behave as if tomorrow mattered. That is the mystery that should not exist. Every living thing on Earth, every bacterium, tree, bird, and human, can trace its lineage back to a single ancestral population. Not a creature with eyes or limbs, not something you could recognize as alive by modern standards, but a biochemical system that learned one radical trick: how to persist. Scientists call it LUCA, the last Universal Common Ancestor, but that name hides the true strangeness of what it represents. LUCA is not just our ancestor. It is the moment the universe stopped being purely physical and became biological. Before LUCA, there was no life to lose. After LUCA, death became possible. That alone should give us pause. Because death only exists once life exists. Fear only exists once survival exists. Meaning only exists once something can end. The moment matter learned how to live, it also learned how to fail, and everything since has been shaped by that tension. We often ask how life evolved, how species changed, how intelligence emerged. But those questions assume something far more fundamental has already been answered. They assume life was inevitable. That it was waiting, somewhere in the chemistry of the universe, ready to emerge as soon as conditions allowed. But the deeper question is far more unsettling. Why did anything start caring whether it continued at all? The early universe had no obligation to produce life. Physics works perfectly well without it. Galaxies form, stars burn, atoms combine and break apart, all without awareness, without memory, without purpose. For billions of years, the universe was exactly that, a vast, unfolding process with no witnesses and no stake in its own future. Life did not improve the universe's efficiency. It did not make it more stable. In many ways, life complicates everything. It creates fragile structures that require constant energy to maintain. It builds patterns that entropy is always trying to erase. From a purely physical perspective, life looks like a mistake, a temporary rebellion against the natural drift toward disorder. And yet, once it began, it never stopped. From the first fragile systems that could barely hold themselves together, life spread into every crack the planet offered. It adapted to boiling water, crushing pressure, toxic metals, and radiation that would shatter modern cells. It survived planetary catastrophes, mass extinctions, and global freezes. It did not aim for complexity. It did not plan for intelligence. It simply persisted. That persistence is what makes the origin of life so disturbing. Because persistence implies preference. Somewhere in the deep past, matter began behaving as if existing was better than not existing. Not consciously. Not intentionally. But functionally. Chemical reactions became organized in ways that favored continuation over collapse. Systems emerged that repaired themselves, copied themselves, and competed for resources, long before there was anything that could think, decide, or feel. The universe did not ask for this. No law of physics demanded it. And yet it happened. Imagine rewinding time to before life began. Not to dinosaurs, not to the first cells, but further back, to a world where Earth was just another hostile rock orbiting an ordinary star. No oxygen to breathe. No soil to grow in. Oceans that were acidic and hot, skies filled with poisonous gases, surfaces constantly reshaped by impacts from space. If you were standing there, if standing were even possible, you would see nothing that suggested a future filled with forests, coral reefs, cities, or minds capable of asking questions. You would see chemistry, motion, chaos. You would not see destiny. And yet, hidden within that chaos was the seed of everything you know. Not because it was meant to be there, but because under the right conditions, chemistry can begin to organize itself in unsettling ways. Molecules can form networks. Networks can stabilize. Stabilized systems can persist longer than unstable ones. And once persistence enters the picture, the rules change. Time becomes relevant. The past starts to matter. Mistakes become opportunities. This is the threshold where the story truly begins, not with a living being, but with a process that refused to reset back to zero. A process that carried information forward, even imperfectly. A process that made the future different from the past. That process is what LUCA represents. Not a single organism swimming alone in a primordial ocean, but a shared inheritance, a biochemical language that every living thing still speaks. The same genetic code. The same molecular machines. The same basic strategies for extracting energy and building structure. You carry that inheritance in every cell of your body. The way your cells copy DNA. The way they produce proteins. The way they store energy. These are not human inventions. They are ancient solutions, refined over billions of years, inherited without interruption from the earliest life on Earth. When you breathe, eat, heal, or grow, you are using machinery that predates animals, plants, and even complex cells themselves. You are animated by processes that were already old when Earth was young. This is not poetic exaggeration. It is biological fact. And it raises a troubling implication. If all life shares a common ancestor, then life only had to begin once. Out of all the possible chemical experiments happening across early Earth, in oceans, hydrothermal vents, tidal pools, and mineral surfaces, only one lineage survived long enough to leave descendants. Everything alive today is part of that single, unbroken chain. Which means the origin of life was not just rare. It was singular. We do not see multiple independent origins of life coexisting today. We see one success story, surrounded by the silence of countless failures. Failed chemistries. Failed systems. Failed attempts that left no descendants and no trace. Life did not flood into existence. It clawed its way into it. And once it succeeded, it reshaped the planet so thoroughly that it erased most evidence of how fragile that beginning truly was. The atmosphere changed. The oceans changed. The surface transformed. The world we see today is a world already conquered by life, a world that hides how close Earth once was to remaining forever sterile. This makes the question even sharper. If life is so resilient now, why was it so hard to start? Why did it take hundreds of millions of years after Earth formed for life to appear? Why did it require such specific conditions, such narrow windows of opportunity? And why, once it began, did it lock into a single shared biochemical framework that all future life would be forced to inherit? These are not just scientific questions. They are existential ones. Because if life is rare, then our existence is fragile beyond comprehension. If life is common, then the universe may be quietly alive in ways we have yet to discover. And if life is neither inevitable nor unique, if it is contingent, precarious, and deeply accidental, then meaning itself becomes something we must confront differently. We are not the universe's goal. We are its consequence. The story of LUCA is not the story of humanity's beginning. It is the story of when matter stopped being just matter. When chemistry began remembering itself. When the universe, for the first time, produced something that could be lost. And from that moment on, everything was at stake. Because once life exists, extinction becomes possible. And once extinction is possible, survival becomes the quiet force shaping all futures. This is where our journey begins, not with creatures or ecosystems, but with a single, fragile transition. A transition so subtle it left no fossils. So ancient it predates all familiar worlds. And so profound that without it, the universe would still be silent. Matter learned how to live. And nothing has been the same since. Before there was life, there was time, but time was not measured in years or seasons. It was an indifferent, sprawling expanse where galaxies collided, stars ignited and died, and planets coalesced from clouds of dust. The universe moved in grand rhythms, entirely unconcerned with anything resembling consciousness. Matter danced, energy flowed, and yet, there was no observer to mark these movements, no voice to ask, why, or how. The cosmos was vast, beautiful, and utterly mute, a theater of motion without audience or purpose. If you could travel back through this emptiness, to the very first moments after the Big Bang, you would see nothing like life. You would see light emerging from chaos, temperatures so high that atoms themselves could not exist, and radiation coursing freely through a young, expanding universe. There was structure forming, yes, but it was mathematical and impersonal. Clouds of hydrogen collapsed under gravity to form the first stars. These stars, burning for millions of years, forged the elements that would one day be essential to life: carbon, nitrogen, oxygen, phosphorus, and sulfur, yet none of these elements carried intent. They were merely the byproducts of nuclear reactions, as indifferent to future life as a stone is to a river. The Earth itself had yet to exist, and yet the seeds of possibility were already scattered. Dust from exploded stars drifted across space, coalescing slowly into the solar nebula that would become our Sun and its orbiting planets. Every particle that would eventually make up our planet had a violent history. Some had been forged in the hearts of stars, others flung across light years in supernovae, and yet all would converge, by chance and physics alone, to create a small, rocky world capable of supporting chemistry, and, eventually, biology. When Earth finally took shape, it was a world of extremes. Molten surfaces churned beneath a turbulent atmosphere of toxic gases, bombarded continuously by asteroids and comets. Oceans were acidic and unstable, their compositions fluctuating with every impact. Life as we understand it today would have been instantly destroyed. And yet, the universe had provided the raw ingredients: carbon for structure, hydrogen for water, nitrogen and phosphorus for information, sulfur for energy, all set upon a stage that seemed perfectly hostile. The paradox was profound: conditions seemed perfectly aligned to create life, but only if matter could somehow overcome chaos itself. In those early moments, the universe and the Earth existed without witness or memory. There were no organisms to adapt, no systems to stabilize, no replicators to persist. Chemistry was happening, but it was aimless, a series of reactions dictated entirely by probability, energy gradients, and the laws of physics. Molecules formed and broke apart as easily as waves crash and retreat upon a shore, leaving no trace of what had passed. And yet, hidden within the laws that governed these molecules, there were rules subtle enough to allow order to emerge spontaneously. Consider a simple amino acid forming in a warm, mineral-rich pond. Alone, it is nothing but a molecule, shaped by chance collisions and chemical affinities. Yet, in the right conditions, these molecules can begin to interact, linking into chains, folding into shapes, and catalyzing new reactions. In these interactions, we can begin to see the first whisper of organization, a faint echo of the persistence that would one day characterize life. It is not yet alive. It has no purpose, no agency, no desire to continue. But it is the beginning of something that might. Time itself, in this pre-life era, was indifferent. Billions of years passed in silence, the slow ticking of cosmic events indifferent to outcomes. Stars burned out, comets crashed, and tectonic plates shifted, yet none of these phenomena carried any preference. Survival was irrelevant because nothing could live or die. Entropy reigned supreme. Matter tended toward disorder, energy flowed from hot to cold, and yet, within this vast expanse of inevitability, tiny pockets of stability began to form. These islands of stability, whether within hydrothermal vents, mineral surfaces, or shallow pools, became the cradles of the improbable. The profoundness of this era is often overlooked. We think of life as ancient, but it is easy to forget just how much of the universe's history passed without a single molecule crossing the threshold into biology. Billions of years of pure physics preceded the first chemistry that could even hint at persistence. This deep emptiness allows us to appreciate how improbable life truly is. Every reaction, every bond, every energy transfer that eventually led to LUCA was a statistical miracle. Each stable molecular assembly was a tiny victory against the relentless tide of randomness. During this time, Earth itself began to prepare for what was to come. Its surface cooled, water condensed into oceans, and minerals formed crystals that could catalyze chemical reactions. Lightning sparked across the skies, radiation penetrated the waters, and tidal forces stirred shallow pools. In this violent, chaotic theater, prebiotic chemistry unfolded invisibly. Organic molecules multiplied, collided, and sometimes formed self-reinforcing networks. These networks were fragile, transient, and vulnerable to destruction, yet they marked the earliest steps toward life, a step that mattered in a universe otherwise unconcerned with matter's fate. It is tempting to imagine this era as quiet or uneventful, but the scale of change was staggering. Stars lived and died, forging heavier elements that were scattered across space, some destined for Earth. Volcanoes reshaped continents that had not yet fully formed. Oceans rose and fell with catastrophic regularity. These forces were indifferent, often destructive, and yet they provided the crucible in which life's first sparks could ignite. Chaos itself was not an obstacle but a medium, shaping molecules and energy flows, forcing chemical systems to adapt or vanish. Amid this backdrop, the stage was set for something extraordinary. Stability and persistence, the hallmarks of life, were beginning to appear not by intention but as a consequence of natural law. Molecules that could catalyze their own replication, even imperfectly, gained an advantage. Networks of chemicals that could resist disruption lasted longer. Information, in the form of molecular patterns, began to persist across time. Here, in this universe that had no reason to favor life, the first threads of continuity emerged, threads that would one day be woven into the fabric of every living cell. What is remarkable about this time is not just the emergence of chemistry capable of life, but the patience of the universe itself. Billions of years passed, during which countless failures occurred. Molecules assembled and disassembled, reactive compounds destroyed one another, energy was wasted. There were no witnesses, no engineers, no shepherds guiding the process. The universe simply allowed processes to continue, and in that allowance, something miraculous happened. By chance, by physics, and by the slow accumulation of improbable events, matter began to acquire a form of memory, not consciousness, but persistence, that would be inherited by every organism to come. When we look at the universe now, it is easy to forget this deep prelude. Galaxies swirl, stars shine, and life teems on one small planet. But all of this rests upon a history so vast, so indifferent, that it is almost unimaginable. The emergence of life is not a natural conclusion of cosmic events, it is an outlier, a delicate thread of order in a universe predisposed to disorder. And yet, within that thread lies the origin of everything we recognize as living. It is here, in this prelude, that we confront the true scale of what LUCA represents. Long before cells, before metabolism, before even RNA, there was a universe poised on the edge of possibility. Chemistry was happening, yes, but it was only the persistent arrangements, those that could survive long enough to influence what came next, that set the stage for life. In this sense, LUCA is not a moment in time, but but a culmination of eons of improbable events, a testament to the quiet persistence of matter that, against all odds, learned to continue. The story of life does not start with a creature or an ecosystem. It begins in this vast, indifferent silence, where the universe itself was patient enough to allow chemistry to flirt with continuity. It is a story that demands awe, not because life was inevitable, but because it was so profoundly unlikely. Every molecule that would eventually be part of LUCA had to survive countless obstacles, from cosmic radiation to planetary chaos, before joining forces in the first system that could truly endure. This realization, that life emerged from a universe that had no reason to produce it, is both humbling and exhilarating. We are the descendants of a single, improbable persistence, born from eons of randomness and silence. Every breath, every thought, every heartbeat is a continuation of that deep cosmic patience. And in understanding this, we can begin to appreciate not only the fragility of life but the astonishing improbability that anything, anywhere, could ever have begun at all. The universe did not intend life. It did not plan it, shape it, or demand it. Yet, through chance, persistence, and the slow accumulation of order from chaos, life became inevitable once it began. And in that inevitability, we see the first traces of a story that will continue for billions of years, a story that begins not with a creature, not with a cell, but with the patient, silent universe learning how to remember itself. Earth was not born gentle. When it first formed from the chaos of dust and molten rock, it was a world so hostile that even the simplest chemical bonds struggled to endure. Lava flowed endlessly, reshaping a landscape that had yet to settle into continents. Gases, carbon dioxide, methane, ammonia, hydrogen sulfide, swirled in a toxic atmosphere. There was no sky to breathe, no ocean to swim, and no soil in which to root. Water existed, but only sporadically, often vaporized by the relentless heat or scalding down in brief, violent storms. The surface was a constantly shifting, searing terrain, bombarded by rocks and ice from space that hammered the young planet with explosive energy. Life, as we imagine it, would have been unthinkable. And yet, within this violence, the planet began to prepare itself. Molten rock gradually cooled, forming primitive crusts. Tiny pockets of water collected in basins, isolated from the surface's chaos. Underwater volcanic vents punctuated the oceans, releasing heat and minerals in streams that could create chemical gradients. Lightning flashed across the sky, delivering energy that could drive chemical reactions. Each of these processes, individually violent and destructive, collectively created the subtle conditions that might one day support life. The planet itself was a crucible, not nurturing but shaping, not gentle but essential. The relentless impacts from space, which might seem only destructive, also played a strange role. Comets and asteroids carried water and organic molecules, seeding the surface with the raw materials of chemistry. Their collisions, while catastrophic, stirred the primordial soup, increasing the chances of molecular interactions. It was a chaotic mixing bowl, and in chaos, there is possibility. The very forces that made the Earth almost impossible to survive also made it chemically interesting. Each impact, each volcanic eruption, each searing lightning strike was a kind of brutal experimentation, shaping the conditions for life without knowing it, without caring. Beneath the ocean's surface, conditions were slightly less hostile. Hydrothermal vents expelled mineral-rich fluids, creating gradients in temperature, pH, and chemical composition. These gradients are crucial because life, at its core, is about managing energy differences. The first chemical systems that would one day evolve into cells likely thrived here, in the margins between extreme heat and the cold abyss, between toxicity and the relative stability of water. It was in these extremes that early chemistry discovered the principles of persistence, a reaction that could sustain itself longer than the surrounding chaos would survive, while others faded into oblivion. Earth's early atmosphere was equally brutal. Oxygen, the very gas that now sustains complex life, was absent. Instead, the air was thick with poisonous gases, suffocating and reactive. Ultraviolet radiation from the sun bombarded the surface without protection. For life as we know it, these were death sentences. Yet, in this hostile world, chemistry continued. Molecules that could endure energy fluxes, that could persist through destruction, began to accumulate. Some formed protective shells, others folded into shapes that were slightly more stable than the alternatives. From such small advantages, persistence slowly emerged, the very principle that would define all living systems. The surface itself was a landscape of extremes. Some regions were scorched barren plains of molten rock, constantly reformed by eruptions and tectonic shifts. Others were deep depressions where water pooled briefly before evaporating. Mountains rose and fell in dramatic episodes of tectonic upheaval, and rifts opened in the crust, releasing heat and minerals from the planet's interior. The environment was inconsistent, unpredictable, and relentless. There was no continuity for life to depend upon, only fleeting stability in small, isolated niches. Those niches, however, would become the incubators of biology's first steps. Even the oceans were not safe. Early water bodies were acidic, enriched with dissolved metals and toxic compounds. Salinity fluctuated wildly as water evaporated and returned through rainfall, itself spurred by the planet's unstable atmosphere. Temperatures were extreme, cycling between near boiling in shallow pools to freezing in isolated pockets during cosmic events. Any chemical system attempting to persist had to be robust to extremes, flexible to change, and capable of exploiting tiny windows of stability. The odds were stacked so high against life that the eventual emergence of LUCA seems almost miraculous, a statistical outlier in an unforgiving world. Yet, it is precisely this hostility that made the first steps toward life possible. Energy was abundant, if unpredictable. Chemical gradients existed everywhere, driven by temperature, pH, and concentration differences. Minerals could catalyze reactions that otherwise would never occur spontaneously. Shifts in tides, volcanic activity, and meteorite impacts constantly churned environments, mixing chemicals in new ways. These dynamic conditions forced protobiological systems to find persistent strategies or perish. In a sense, the universe was teaching chemistry how to resist entropy, through relentless challenge. Consider the paradox: a world almost impossible to inhabit became a laboratory for life. The very elements that threatened destruction, heat, radiation, acidity, heavy metals, also provided energy and catalytic surfaces for molecules to organize. The extremes created gradients that could be harnessed. The violence forced redundancy. Only the most stable reactions and molecules survived the constant upheaval, forming the foundation for increasingly complex networks. In the absence of life, the planet's brutality was a teacher, shaping what would one day become living systems. Life, when it finally appeared, would do so as a defiance of this relentless environment. The earliest chemical systems were fragile, ephemeral, and entirely dependent on narrow conditions. Any deviation in temperature, chemical concentration, or energy input could destroy them. Yet, some systems endured long enough to influence the next cycle, and then the next. Slowly, persistence became encoded in chemistry itself. The violent crucible of early Earth was not only shaping molecules, but instilling in them the principle that defines all life, the drive to continue, no matter the odds. Even in the modern imagination, it is difficult to grasp the intensity of this early world. Today, life seems everywhere, adaptive, resilient. But the first steps toward biology were precarious. Hydrothermal vents might provide heat and minerals, but they were also corrosive and toxic. Shallow pools might concentrate molecules, but they were vulnerable to evaporation or cosmic impacts. Life's precursors existed in a world that could annihilate them instantly. Every surviving molecule, every self-replicating system, was an extraordinary triumph of persistence in a planet determined to destroy. And yet, from this chaos emerged the first whispers of life's organizing principle: separation from the environment. The earliest protocells likely formed protective boundaries, isolating chemical reactions from destructive influences. Within these microscopic fortresses, molecules could persist, replicate, and interact without immediate annihilation. These boundaries were more than physical structures, they were the first acknowledgement that persistence required distinction. Survival meant defining oneself against a hostile external world, a principle that continues in every living cell today. The early Earth, with its searing oceans, toxic skies, and relentless bombardments, might seem inhospitable to life, yet it was precisely the adversity of this environment that made persistence meaningful. Without challenge, there can be no selection. Without destruction, no system can demonstrate endurance. The violence of Earth's formative years was not merely background, it was an integral part of life's emergence. Each catastrophic event, each unpredictable fluctuation, each brutal chemical interaction contributed to the shaping of LUCA and every lineage that would follow. In the end, the lesson is profound: life did not emerge in spite of early Earth's hostility. It emerged because of it. The planet's extreme conditions forced chemistry into patterns, energy flows into persistence, and molecular networks into resilience. What we now recognize as life, self-replicating, adaptive, and enduring, is the product of a planet that seemed designed to prevent it. The improbability of this emergence underscores the fragile miracle of existence. LUCA, and all that followed, is the universe's quiet defiance of entropy, born in a world that should have allowed nothing to endure. In the molten chaos of early Earth, something remarkable began to happen. Not life as we recognize it, not even close, but the first hints that matter could behave in ways that hinted at persistence, at organization, at memory. Chemistry, which had been following only the indifferent laws of physics, started to explore possibilities that were, in a sense, creative. Simple molecules, colliding in oceans, puddles, and hydrothermal vents, began forming combinations that could sustain themselves just a little longer than the chaos surrounding them. For the first time, reactions were not merely happening, they were beginning to favor continuity. At first, these were tiny, almost imperceptible advantages. One amino acid might bond slightly more stably to another. One lipid might form a small, protective bubble, keeping certain reactions isolated from destruction. A chain of nucleotides could form by chance, holding a fleeting record of its own structure. Alone, each of these events was insignificant, easily destroyed by heat, radiation, or turbulence. But together, repeated countless times over millions of years, these small moments of stability became the building blocks of something extraordinary: protobiological organization. The oceans themselves became a laboratory. Deep beneath the boiling surface, hydrothermal vents spewed mineral-rich fluids into the surrounding water, creating strong chemical gradients, differences in temperature, acidity, and composition, that could be harnessed by molecules capable of exploiting them. In these microenvironments, energy flowed not randomly but directionally, creating opportunities for molecules to persist. Molecules that could catalyze their own formation, or protect themselves from degradation, began to survive slightly longer than others. Gradually, these molecular systems became more complex, networks of interactions emerging from the chaos, driven entirely by chemistry, yet hinting at the first principles of biology. Consider the amino acids, the simple chains of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Alone, they are unremarkable. But when conditions are right, they link to form peptides, which fold into shapes capable of catalyzing reactions, tiny molecular machines. Some of these peptides may have helped form protective membranes, while others may have accelerated reactions that would otherwise be too slow to persist. This is chemistry beginning to take on the character of life, not conscious, not purposeful, but increasingly capable of sustaining itself against the surrounding disorder. Lipids, molecules that naturally form bilayers in water, played a pivotal role. In isolation, a lipid is just a simple molecule. But together, lipids spontaneously assemble into vesicles, tiny bubbles that can encapsulate other molecules. These vesicles are the ancestors of the first cell membranes, creating a boundary between internal chemistry and external chaos. Within these primitive compartments, molecules could interact in a controlled environment, reactions could be preserved for longer, and energy could be harnessed more efficiently. Without these compartments, the first molecular networks would have been scattered, diluted, and destroyed by the surrounding environment. Encapsulation was the first lesson matter learned. Survival requires distinction. RNA, a molecule that would later dominate the early stages of life, exemplifies this transition. Unlike DNA, RNA can act both as an information carrier and as a catalyst. In the prebiotic world, RNA or RNA-like molecules could copy themselves, albeit imperfectly, and catalyze chemical reactions. These self-replicating systems, fragile and error-prone as they were, represent the first moment where chemistry gained memory. Information began to persist. Patterns could now be repeated, modified, and passed forward. In the prebiotic soup, RNA molecules that could replicate more efficiently lasted longer, shaping the chemical environment for the next cycle of reactions. Errors, paradoxically, became a driving force. While perfect replication would preserve a single form, errors introduced variation. Some mistakes were destructive, and the molecules disappeared. Others accidentally produced new structures capable of exploiting their environment in ways that previous forms could not. In this way, chance and instability became essential allies in the transition from chemistry to biology. Variation allowed experimentation on a molecular scale, and the fittest reaction survived. The principle that would later govern evolution, the survival of the most persistent, was quietly at work long before cells existed. Energy flow was another crucial element. Life is, at its core, a system of energy management. Early chemical systems needed gradients, differences in temperature, acidity, or chemical concentration, to drive reactions forward. Hydrothermal vents, volcanic activity, and lightning provided constant sources of energy, creating pockets where molecules could capture and harness these differences. Molecules that could take advantage of these energy flows survived longer, creating a feedback loop. Persistence enabled energy capture, which in turn enhanced persistence. Slowly, matter began to behave as if it had a preference, a bias toward continuity. Minerals in the environment also acted as catalysts, accelerating reactions that would otherwise be impossible. Clay, iron-sulfur compounds, and other surfaces provided structure, aligning molecules in ways that increased the likelihood of bonding and replication. These surfaces acted like scaffolds, giving structure to otherwise chaotic chemistry. Without these physical frameworks, the delicate dance of prebiotic molecules might have dissipated entirely. The environment, hostile as it was, became both challenge and enabler, shaping chemistry into forms that could eventually persist on their own. Isolation, repetition, and interaction gradually created networks of molecules that were more than the sum of their parts. These networks were not conscious, but they exhibited emergent properties, patterns that persisted, replicated, and adapted. They were protobiological in every sense except one: they were not alive. Yet, they carried the potential for life. The line between chemistry and biology began to blur. Molecules no longer simply reacted, they began to organize, store information, and influence their own futures. In the chaotic, toxic oceans of early Earth, persistence had become a tangible force. It is remarkable to consider how improbable this process was. Billions of years of relentless bombardment, chemical destruction, and environmental fluctuation might easily have erased every nascent molecular network. Yet, against overwhelming odds, certain patterns endured. Persistence became a property encoded in matter itself. The molecules that survived were those capable of resisting destruction, of harnessing energy, of storing information, even if only temporarily. These systems, fragile and provisional, formed the first hints of the biological principle that would govern all life: continuity in the face of chaos. As this prebiotic chemistry unfolded, the stage was quietly being set for LUCA. By accumulating information, forming boundaries, harnessing energy, and surviving errors, these molecules established the essential features of life long before a cell, an organism, or a recognizable ancestor appeared. The first sparks of biology were born not from intention, but from the interplay between order and chaos, persistence and destruction, energy and matter. Chemistry had learned to act as if it mattered. And in this quiet, invisible transformation, the universe had crossed a boundary it had never crossed before. Matter, for the first time, began to behave in ways that hinted at life. Reactions were no longer purely random. Networks could persist. Information could be stored and transmitted. Energy could be harnessed. Stability could be selected over instability. The transition was subtle, almost imperceptible, yet profound. The first steps from matter to life had been taken, in silence, long before anything resembling a creature existed. The lesson of this era is both simple and astonishing: life did not emerge fully formed. It was a gradual, improbable, and delicate accumulation of molecular persistence, enabled by a hostile environment that both destroyed and facilitated it. Chemistry had learned new tricks, organizing itself into networks capable of enduring, influencing, and replicating. And in doing so, it set the stage for everything that would come after: metabolism, cells, DNA, RNA, and eventually, the last universal common ancestor. In the chaotic oceans and mineral-rich pools of early Earth, life's first premonitions arose, not as living beings, but as matter daring to persist. And it is here, in this subtle, invisible emergence, that we begin to glimpse the profound mystery of our origins, that everything alive today traces back to this quiet revolution, when chemistry learned to defy indifference, to organize, and to endure. Out of the chaos of early Earth, a faint, almost imperceptible pattern began to emerge. The molecules that had persisted through random collisions, extreme heat, and constant bombardment began to interact in ways that suggested more than chance. These interactions were fragile, fleeting, and easily destroyed, yet they hinted at a principle that would define all living systems: organization. Life had not yet arrived, but the first whisper of it, the earliest experiments in structure, continuity, and self-preservation, was taking place on a scale invisible to the naked eye. Imagine a shallow pool near a volcanic vent. The water is acidic, the temperature fluctuates wildly, and minerals coat every surface. Within this pool, lipids and amino acids interact, forming rudimentary compartments. These protocells, fragile as soap bubbles, provide an isolated space where chemical reactions can occur with reduced interference from the surrounding chaos. Encapsulation allows persistence. Molecules trapped inside a compartment have a higher chance of interacting repeatedly, increasing the likelihood that certain chemical networks will endure. Without such separation, every reaction would be exposed to destruction, dissipating into the surrounding ocean. These early compartments represent the universe's first experiments in selfhood, the idea that survival requires defining an inside against an outside. Within these primitive enclosures, networks of molecules begin to cooperate. Certain molecules catalyze reactions that produce more of themselves or protect other molecules from degradation. These are not conscious decisions but emergent behaviors driven by chemical laws. Persistence becomes self-reinforcing. Molecules that survive longer are more likely to participate in future reactions, creating feedback loops that encourage stability. This is the first lesson of organization: continuity requires interaction, and interaction requires structure. The universe, indifferent as it is, allows systems that can maintain themselves to persist, and gradually, the first building blocks of biological organization take shape. Energy gradients in these early environments become essential. Hydrothermal vents, tidal pools, and mineral-rich surfaces provide constant flows of energy. Differences in temperature, acidity, or chemical composition. Molecules that can exploit these gradients gain an advantage, persisting longer than those that cannot. Even in these primitive conditions, we see the precursor to metabolism, the harnessing of energy to maintain organization. This is not life yet, but it is life's first strategy, an early negotiation with the forces of entropy that threaten to erase all order. RNA or RNA-like molecules begin to play a critical role. Their dual capacity for information storage and catalysis allows them to act as both record keepers and functional agents within these protosystems. Some strands fold into shapes that accelerate their own replication or catalyze other reactions, effectively learning how to persist longer than other molecules. These early molecular systems exhibit the first signs of heredity, however rudimentary, patterns that can be copied, even imperfectly, giving rise to variation. Within these compartments, chemistry is no longer purely reactive. It has memory, albeit primitive, and the first principles of natural selection are silently at work. Variation, introduced by imperfect replication or chemical instability, becomes a driving force. Some molecular arrangements are destroyed almost immediately, while others survive, adapt, and influence subsequent reactions. These small differences are the ancestors of evolution. In the absence of life as we know it, chemical systems are already subject to selection pressures. Persistence favors certain structures, energy efficiency favors others. Through countless cycles, molecules that can survive and reproduce even slightly more effectively begin to dominate their microenvironments. Complexity emerges incrementally, guided entirely by survival, not by design. The boundary between inside and outside becomes increasingly significant. Molecules confined within a compartment are protected from destructive forces, allowing more intricate networks to form. This separation is the earliest recognition that life depends not only on the molecules themselves but on their context. Stability emerges as a relational property. A molecule's persistence depends on the environment and its interactions with neighbors. From this perspective, organization is a dance between the internal and external, a negotiation between protection and exposure, between persistence and entropy. These proto-organisms, though microscopic and ephemeral, also begin to exhibit rudimentary communication with their surroundings. Chemical signals diffuse across compartment boundaries, influencing the behavior of neighboring molecules. Gradients of pH, ions, or other compounds provide information about local conditions. While far from conscious awareness, these interactions represent the first form of environmental feedback, a primitive awareness encoded in chemistry. Systems that respond appropriately to environmental cues persist, while those that do not fade away. This feedback loop, so central to life, is already emerging in the earliest molecular networks. Stability, persistence, and interaction create the foundation for increasingly complex structures. Molecules that survive longer accumulate, forming networks capable of sustaining more intricate reactions. Some compounds catalyze the formation of others, creating chains of dependencies. Others protect vulnerable molecules, acting as early molecular caretakers. Even at this microscopic scale, we see the roots of cooperation, a fundamental principle that will shape life from its earliest cellular forms to complex organisms. It is here, in these humble compartments and chemical networks, that the first principles of life are encoded: separation from the environment, the harnessing of energy, the replication of information, and the ability to respond, however rudimentarily, to conditions. These are not just chemical curiosities, they are the foundation of persistence, the first steps towards something that can survive long enough to evolve. Life has not yet appeared, but matter is beginning to behave as if it intends to continue, to maintain itself, to resist the inevitable pull of disorder. Environmental variability remains a powerful force. Temperature swings, chemical fluctuations, and energy pulses continually threaten these fragile systems. Yet, this instability is not merely destructive, it is selective. Only molecular arrangements capable of withstanding disruption survive, and those that survive shape their surroundings for future reactions. This interplay between chaos and order is the crucible in which life's organizing principles are forged. The first whispers of organization are inseparable from the challenge of survival in a hostile world. Over countless generations of chemical cycles, these proto-organisms develop rudimentary mechanisms to capture energy more efficiently, to store information more reliably, and to protect themselves more effectively. Each incremental improvement is subtle, often temporary, and almost certainly obliterated countless times before succeeding. Yet persistence is cumulative. Molecules that endure long enough influence the next generation, leaving behind patterns that gradually increase in complexity. In this way, organization grows organically from the laws of chemistry and physics, without any guiding hand, as if matter itself is learning to persist. The universe, indifferent as it is, allows for this emergent order. It does not intervene, it does not plan, and it does not judge. Yet, in the interplay of chance, repetition, and selective persistence, something remarkable arises, the first signs of what we might call proto-life. These systems are neither alive nor conscious, but they exhibit all the hallmarks of persistence that will define life once it appears. Boundaries, energy utilization, replication, variation, and interaction, all emerge from the relentless interplay of molecules and environment. From these first whispers of organization, every living system to come will inherit a principle: survival is not passive. It requires structure, feedback, and the ability to influence one's own persistence. Encapsulation, catalysis, energy flow, and memory, these are the lessons learned in the prebiotic soup. LUCA, the last universal common ancestor, will carry these lessons billions of years later. And so, from the invisible chaos of early Earth, the stage is set, matter has begun to behave as if it matters, and the first real steps toward life, fragile and impermanent though they may be, are underway. Long before DNA existed, before proteins became the architects of cells, there was RNA, a molecule that carried the first glimmers of biological memory. In the prebiotic world, RNA or RNA-like molecules were unique. They could store information in their sequences, and at the same time catalyze chemical reactions. They were not alive in any sense we would recognize, yet they had a property that would become foundational for life: the ability to influence their own persistence. They were both record keepers and actors, writing a history in chemical bonds while subtly shaping the future of the molecular environment around them. Imagine a pool of water, mineral-rich, fluctuating in temperature and chemical composition, with RNA strands forming, folding, and breaking apart. Most molecules fail almost immediately, destroyed by heat, radiation, or chemical instability. Some survive slightly longer, merely by chance. A particular strand folds in such a way that it catalyzes the formation of another strand like itself. For a brief moment, a cycle of replication emerges. These are fragile, imperfect copies, but they carry information forward. Errors creep in naturally, altering sequences and creating variation. Some errors are catastrophic, but others open new possibilities, generating molecules that can catalyze reactions that no previous strand could. In this process, the very first principle of evolution, variation, persistence, and selection, is quietly at work long before life exists. RNA molecules represent a kind of memory before meaning. They store information chemically, holding a record of past reactions and folding patterns. This memory is not conscious, but it is functionally similar to what life will later exploit: the ability to influence the future based on past configurations. A molecule that folds more stably survives longer, interacts more effectively, and reproduces more frequently than its less stable counterparts. In this way, RNA allows the environment to shape chemistry over time, and in turn, the chemistry begins to shape its environment. What begins as a chance folding pattern becomes a persistent influence, a trace of continuity in a world otherwise ruled by chaos. Energy remains the critical currency of persistence. The hydrothermal vents, volcanic pools, and mineral surfaces of early Earth provide gradients, differences in temperature, chemical potential, and acidity, that drive reactions forward. RNA molecules that can exploit these gradients, even indirectly, gain an advantage. Some catalyze reactions that extract energy from chemical differences, others stabilize local environments to reduce destructive fluctuations. Through repeated cycles, these molecular systems demonstrate the first rudimentary metabolism. Energy is not simply consumed, it is harnessed to maintain organization and to persist. Errors, while destructive, are essential. Perfect replication would produce nothing but sameness, a static system incapable of exploration. Imperfections allow novelty. Slight variations in RNA sequences can lead to new catalytic properties, new ways of harnessing energy, or new interactions with neighboring molecules. Some variations fail, and those strands vanish. Others survive, creating cascades of chemical possibilities that gradually increase the complexity of the system. In this way, error is not failure, it is experimentation, the first mechanism of innovation in the prebiotic world. Compartmentalization becomes increasingly significant. RNA strands trapped within lipid vesicles or mineral cavities interact more effectively and are shielded from destructive forces. These boundaries provide a controlled environment, allowing certain reactions to persist longer than others. Within these compartments, RNA molecules begin to cooperate indirectly. Some catalyze reactions that produce components needed by others, forming the earliest networks of interdependence. Cooperation at this scale is emergent, unintentional, yet crucial. Even at this microscopic, chemical level, the foundations of communal survival are laid. This is a world defined by fragility. Strands are short-lived, vesicles rupture, temperature spikes can destroy entire microenvironments. Yet, persistence matters. Molecules that survive long enough to replicate influence subsequent generations. The interplay between destruction and survival creates a selection pressure without consciousness, without purpose, but with profound consequences. Only the systems capable of enduring instability, exploiting energy flows, and reproducing, imperfectly though they may, leave traces that echo into the future. Consider the paradox: these RNA molecules are not alive, yet they exhibit properties we associate with life. They store information. They reproduce, albeit imperfectly. They harness energy. They respond to their environment in ways that enhance persistence. They vary, and variation enables selection. And yet, they are merely complex chemistry, subject to the same physical laws that govern every molecule in the universe. Life, as we will come to know it, will emerge from the continuation and refinement of these principles. Time, in this context, begins to have meaning. Before RNA, chemical reactions occur and dissipate with no cumulative effect. With RNA, past configurations influence future possibilities. Molecules that have survived long enough leave patterns that guide subsequent reactions. The first threads of continuity emerge. The environment begins to remember the systems that persist, not consciously, but through the presence of molecules capable of catalysis and replication. Memory is chemical, not cognitive, yet it is foundational. In some ways, RNA molecules act as the first bridge between chemistry and biology. They embody a duality, reactive yet preservative, transient yet persistent. They demonstrate that information can be encoded, transmitted, and modified, even in a world with no cells, no metabolism, and no life. These early molecules teach an essential lesson: survival is not a property of individual atoms, but of networks capable of maintaining themselves through time, exploiting energy, and transmitting structure. The RNA world is also a study in contingency. The precise conditions that allowed RNA to form and persist were incredibly specific. Variations in temperature, pH, mineral composition, and chemical concentration could destroy nascent RNA systems instantly. Yet, pockets of stability existed, tiny havens where molecules could fold, catalyze reactions, and replicate. These niches allowed the first persistent molecular networks to emerge. Without them, life might never have begun. The universe allowed persistence, but only narrowly, only fleetingly, and only where the conditions permitted. From these RNA molecules, all life inherits a profound legacy. The principles they embody, information storage, replication, catalysis, variation, and selection, form the backbone of biology. DNA, proteins, cells, and ultimately complex organisms all rely on these foundational mechanisms. In a very real sense, every living system today is an elaboration of the strategies first developed in the RNA world, refined over billions of years, but rooted in the same chemical principles. What is extraordinary is that all of this occurs without consciousness, intention, or purpose. The RNA world is neither alive nor aware, yet it exhibits persistence, memory, and a protoform of evolution. It demonstrates that life's essential principles can emerge spontaneously from matter under the right conditions. The universe, indifferent to its own creations, allows chemistry to explore the possibilities of persistence, and in doing so, lays the groundwork for the most improbable event in cosmic history, the origin of life itself. It is here, in this delicate, invisible world, that we glimpse the first genuine step toward life. RNA molecules, fragile yet persistent, reactive yet preservative, mark the transition from mere chemistry to systems capable of continuity. They are the first to carry memory, to influence their own persistence, and to enable the accumulation of complexity over time. Through them, matter begins to behave as if it has a future, even when no one is there to observe it. In the RNA world, memory precedes meaning. Replication precedes identity. Persistence precedes consciousness. And yet, these humble molecules are the ancestors of everything we call life. From these ephemeral strands arose the first systems that could endure, adapt, and evolve. From them, over billions of years, would emerge LUCA, the last Universal Common Ancestor, the first organism to embody the lessons of RNA and carry them forward into every cell on Earth today. The RNA world teaches us something profound. Life is not a miracle because it is beautiful or intelligent, but because it is persistent. In the midst of chaos, fragility, and destruction, molecules learned how to endure. They encoded the first memory of survival, and in doing so, set the stage for everything that would follow: cells, metabolism, ecosystems, and eventually, humanity itself. Memory before meaning was the universe's first step toward life, a quiet revolution written in chemical bonds long before there was anyone to read it. In the delicate world of early RNA molecules, errors were inevitable. Replication was imperfect, folding was inconsistent, and chemical instability constantly threatened these fragile systems. At first glance, these errors seem purely destructive: a strand misfolds and is destroyed, a replication cycle fails, a compartment collapses under thermal stress. Yet within these mistakes lay the seeds of life's most profound innovations. Without error, there would be no variation, no experimentation, no opportunity for molecules to discover solutions to the challenges imposed by their hostile environment. Chaos, it turns out, was not only the backdrop of persistence, it was the engine of creativity. Consider a single RNA strand copying itself within a tiny vesicle. Every time it replicates, slight mismatches occur. Some substitutions break the molecule entirely, causing it to dissolve into the surrounding medium. Others, however, might fold differently, enabling the molecule to catalyze a reaction more efficiently or resist degradation slightly better. This is the first echo of evolution in action: error is filtered by persistence. The molecules that survive, even by the slimmest margin, carry their modifications forward, influencing the chemistry of the next cycle. In the RNA world, survival is the measure of success, and error is the currency of change. Errors also introduced novelty into molecular systems. A single misfolded RNA strand might catalyze a reaction that no previous molecule could, producing new compounds or stabilizing previously unstable reactions. In some cases, these new compounds create feedback loops, enhancing the overall stability of the molecular network. What begins as a chance deviation can ripple outward, shaping an entire microenvironment of chemical interactions. In this sense, errors are not failures, they are opportunities for innovation, tiny experiments carried out at the scale of molecules, invisible but transformative. The interplay between destruction and novelty creates an emergent principle: selection. Molecular systems that persist do so not because they are perfect, but because they are resilient to error. Fragility and flexibility coexist. Some RNA strands fold rigidly and are destroyed by slight environmental fluctuation, others fold dynamically, tolerating mistakes, and thus persist longer. This principle, repeated endlessly across countless molecular generations, establishes the earliest form of natural selection. The first life did not compete consciously, but persistence and error produced an invisible battlefield where only the most resilient systems endured. Errors also allow systems to explore chemical space in ways perfect replication cannot. In a world dominated by chance, variation is the only mechanism for discovery. Each misfolded strand, each incorrect replication, represents a tiny deviation from the norm, a chemical experiment unconsciously testing new strategies for survival. Over time, these experiments accumulate. Some fail immediately, disappearing without a trace. Others succeed, creating new catalytic possibilities, stabilizing interactions, or new ways to harness energy. The early RNA world was a laboratory of infinite possibility, conducted not by intent but by necessity. One of the most profound effects of error is its role in generating networks of interdependence. A misfolded RNA might catalyze the formation of a lipid molecule, which in turn stabilizes other RNA strands within a vesicle. Another error might produce a small peptide that enhances catalytic efficiency. Over countless cycles, these chance interactions produce cooperation at a molecular level. Early chemistry, driven by error, begins to resemble a primitive ecosystem, networks of molecules relying on one another, persisting collectively, adapting indirectly through the consequences of chance. Errors also accelerate complexity. In a static, perfectly replicated system, no new interactions arise, and chemical evolution stagnates. Mistakes, by contrast, create variation that allows molecules to explore previously inaccessible pathways. A single alteration might enable a strand to interact with a new substrate, stabilize a membrane, or catalyze a reaction that generates energy more efficiently. These small innovations, filtered by survival and persistence, accumulate over time. From the chaos of error, complexity emerges, not suddenly, but gradually, invisibly, inexorably. Energy utilization is shaped by error as well. Some molecular variations allow more efficient harnessing of chemical gradients, extracting energy from environmental differences that less adapted molecules cannot exploit. Others stabilize reactions that would otherwise dissipate rapidly. The ability to capture, store, and redirect energy becomes a selective advantage, enhancing persistence. In this sense, error is the engine that drives the emergence of metabolism, long before the first cells or recognizable organisms exist. Energy management and error management become intertwined, forming the biochemical foundation of all future life. Compartmentalization amplifies the consequences of error. Within vesicles or mineral cavities, successful molecules persist and interact, while failures are isolated and removed from the larger system. Errors that confer an advantage within these microenvironments can influence local chemical dynamics, producing cascading effects. Some vesicles collapse, taking their molecules to destruction, others survive, enriched with more resilient, innovative chemical networks. Selection occurs at multiple scales, individual molecules, microcompartments, and networks of interactions. Error, far from being random destruction, is the architect of these hierarchies of persistence. Environmental fluctuation interacts with error to produce adaptive feedback. Temperature spikes, pH changes, and chemical fluxes destroy unstable molecules, but those capable of tolerating or exploiting change survive. Mistakes that allow molecules to endure stress become encoded in subsequent generations. Chemical systems begin to exhibit rudimentary adaptability, adjusting to the environment without any intelligence or foresight. The combination of error, persistence, and environmental pressure lays the groundwork for evolution, long before genes, proteins, or cells exist. It is worth reflecting on the paradox: without error, life could not exist, yet errors threaten to destroy life at every turn. Early RNA molecules existed in a delicate balance between fragility and innovation. Each error was a gamble, each replication a test. Most attempts failed, leaving no trace. But the few that survived set in motion a chain of consequences that would echo across billions of years. From these tiny, invisible missteps emerged the molecular principles that underpin all living systems: replication, variation, selection, energy management, and interdependence. The RNA world, shaped by error, demonstrates that persistence is not passive. To endure, a system must navigate instability, exploit opportunity, and respond to the consequences of its own imperfections. Error is not merely tolerated, it is essential. Every mutation, misfold, or misreplication is a step toward complexity, an experiment in survival, a lesson in resilience. LUCA, when it finally appears billions of years later, inherits this legacy of error-driven innovation, embodying the principles forged in the crucible of prebiotic chemistry. Errors also underscore the improbability of life. Each advantageous deviation had to survive countless destructive events: thermal fluctuations, chemical breakdown, environmental instability. The survival of a single beneficial error is a statistical miracle. The accumulation of multiple errors that increase persistence is exponentially more improbable. Yet, against the relentless odds of a hostile planet, these improbable events occurred, quietly, invisibly, and repeatedly, until the first molecular networks capable of true continuity emerged. The lesson of errors is both humbling and profound. Life is born not from perfection, but from resilience. Fragility and instability are not obstacles, they are the conditions under which innovation occurs. Early molecular systems that could endure mistakes, harness energy, and exploit opportunities became the ancestors of all living systems. The RNA world teaches us that survival depends not on flawlessness, but on the capacity to navigate imperfection, to turn chance into continuity, and to use error as a vehicle for persistence. In this unseen world, errors were not random destruction, they were the architects of life's first strategies. Each misstep, each misfold, each replication mistake was a lesson in endurance, a test of persistence, and a quiet revolution in chemical organization. From these errors emerged the first whispers of evolution, the first networks of interdependence, and the first principles of metabolism, replication, and adaptation. Without them, LUCA would never have existed, and the chain of life stretching to the present day would never have been forged. In the shadowed depths of early Earth, where hydrothermal vents spewed mineral-rich fluids into the acidic oceans, the first whispers of metabolism began to take shape. Life, in its earliest chemical form, was not yet recognizable as we know it, but it had discovered the critical principle that would define its persistence: the ability to harness energy. Survival depended not merely on structure or replication, but on the capacity to capture, store, and redirect energy to maintain organization in the face of relentless entropy. These first proto-metabolic systems were crude and fragile, yet they marked a profound shift in the behavior of matter itself. In these turbulent oceans, energy flowed abundantly but chaotically. Temperature gradients radiated from hydrothermal vents, chemical concentrations fluctuated violently, and reactive compounds constantly formed and broke apart. Most molecules were destroyed instantly, consumed by heat or broken apart by chemical instability. Yet some molecular arrangements began to exploit these energy flows, catalyzing reactions that could sustain themselves longer than the surrounding chaos. Persistence became a function of energy management, the earliest hint of what we would later recognize as metabolism. Consider a network of molecules forming within a mineral-rich pocket near a vent. Some molecules catalyze the production of others, creating a chain reaction that produces energy-rich compounds. Others facilitate reactions that stabilize local concentrations, allowing sensitive molecules to survive longer. These interactions are entirely chemical, devoid of consciousness or intent, yet collectively they create a system capable of maintaining itself. Energy is not consumed passively, it is captured and directed toward persistence. This is the first principle of metabolism: organization requires energy, and energy enables survival. Hydrothermal vents play a critical role in this process. They provide continuous flows of reduced compounds, such as hydrogen sulfide, and gradients in temperature and pH, which early molecules could exploit. A simple chemical system that can harness these gradients gains a subtle but decisive advantage. Over countless cycles, molecular arrangements that efficiently capture energy persist, while those that cannot are destroyed. Gradually, these systems become increasingly complex, forming networks that resemble primitive metabolism. In essence, energy flows sculpt chemistry into the first tentative forms of life. Compartmentalization amplifies the effect. Molecules trapped within lipid vesicles or confined by mineral surfaces interact more efficiently, capturing energy and directing reactions in controlled spaces. These compartments reduce interference from destructive environmental forces, allowing reactions to persist long enough to reinforce themselves. Within these tiny domains, protometabolic networks can establish feedback loops. Energy captured in one reaction fuels another, which in turn sustains the first. Encapsulation is not just protection, it is the enabling condition for sustained chemical organization. These early metabolic systems are astonishingly adaptive, despite their simplicity. Fluctuations in temperature, chemical composition, and energy availability act as a form of selection. Molecular arrangements that can tolerate instability, exploit energy surges, or stabilize their local environment are more likely to persist. Error and variation remain central. Slight changes in molecular structure can improve energy capture or stability. Over countless generations, systems capable of managing energy more efficiently accumulate, gradually increasing the complexity and robustness of protometabolic networks. Consider the role of catalysts in this early energy economy. Certain molecules accelerate reactions that generate high-energy compounds, such as simple phosphorylated molecules or redox-active intermediates. These compounds, in turn, fuel other reactions, creating chains of energy transfer that resemble the rudimentary steps of modern metabolism. Minerals such as iron-sulfur clusters and clay surfaces catalyze additional reactions, providing scaffolds that enhance stability and increase efficiency. In this way, the environment itself participates in the shaping of early metabolism, guiding chemical systems toward persistence. The principle of energy gradients extends beyond hydrothermal vents. Shallow tidal pools, evaporating and refilling with mineral-rich water, create fluctuating chemical potentials that can drive reactions. Temperature cycles from day to night, or from volcanic activity, provide energy differentials that protometabolic systems exploit. Lightning and ultraviolet radiation intermittently deliver bursts of energy capable of initiating novel reactions. These dynamic environments serve as natural laboratories, constantly challenging chemical networks and favoring systems that can harness energy and maintain organization. As these networks grow, interactions between molecules become increasingly interdependent. A molecule that captures energy efficiently may produce intermediates that stabilize other molecules. Another molecule might catalyze reactions that replenish energy carriers or repair fragile structures. Even in the absence of life, cooperation emerges as a principle. Systems that work together, chemically, have a higher chance of persistence. Complexity is a product of both energy flow and interdependence, the first inklings of ecological dynamics at a molecular scale. Persistence, energy, and compartmentalization converge to produce protocells, predecessors of the first living cells. These are not yet life as we recognize it, but they exhibit crucial features. They maintain internal organization, harness energy from their surroundings, and replicate, imperfectly but persistently. Membranes isolate reactions from destructive fluctuations, catalytic networks harness gradients, and replication mechanisms propagate successful configurations. These protocells represent the threshold where chemistry begins to behave like biology, where matter begins to act as if it has a future. The emergence of metabolism is also a lesson in improbability. Energy is abundant, but capturing and redirecting it reliably requires precise conditions and networks of molecules capable of exploiting gradients. The odds of forming a functional protometabolic system are vanishingly small, yet the relentless persistence of molecular interactions and environmental cycles allows these systems to arise repeatedly. Over time, selection favors those networks that maximize energy efficiency and resilience, shaping increasingly robust chemical systems capable of sustaining life. Errors and variation remain central. A slight alteration in a catalytic RNA, a misfolded peptide, or a modified lipid can enhance energy capture, stabilize reactions, or improve compartmental integrity. These accidental innovations are preserved through persistence, providing the foundation for gradual refinement of protometabolic networks. In this way, energy management and error-driven adaptation are intertwined, creating a feedback loop that drives the emergence of complexity long before the first true cell exists. By the time LUCA emerges, these principles, energy capture, compartmentalization, catalytic networks, and error-driven adaptation, are fully encoded in the architecture of life. Every cell today inherits the legacy of these first metabolic systems, the chemical strategies that allowed molecules to persist in a hostile, dynamic environment. Life, in essence, begins with energy, the ability to resist chaos by capturing, storing, and redirecting the flows of the universe in order to endure. The lesson is profound. Metabolism is not a trivial property of life, it is the foundation of persistence itself. Without the ability to harness energy, early molecular systems would have dissipated instantly into the entropy of their environment. Energy is what allows molecules to maintain order, reproduce, and influence their surroundings. It is the thread that links chance chemistry to the first living cells, the invisible force that makes persistence possible in a world designed to destroy it. In the emergence of metabolism, we witness the universe teaching matter how to survive. Chemical systems learn to organize, capture energy, and respond to instability. They form compartments, create networks, and exploit gradients. Errors and variation introduce innovation. Persistence is rewarded, fragility eliminated. In these invisible, ancient processes, the stage is finally set for the first true organisms, for the last universal common ancestor, for life itself to step into the history of the planet. In the chaotic seas of early Earth, where hydrothermal vents hissed and mineral-laden waters surged, the first compartments began to take shape. These were not cells in the sense we understand today, but they were the first attempts at creating boundaries, delicate, fragile, and ephemeral, yet vital for the persistence of nascent chemical networks. Encapsulation, the act of separating the internal world from the external chaos, was a revolutionary principle. It allowed molecules to interact, stabilize, and persist without being immediately destroyed by the violent environment surrounding them. For the first time, matter could claim a private space in which the rules of survival could operate more reliably. Lipids, with their natural tendency to form bilayers in water, were the architects of these first compartments. Alone, a lipid is a simple molecule, but in water, lipids spontaneously assemble into vesicles, tiny, enclosed spheres that can trap other molecules inside. Within these vesicles, reactions that would otherwise dissipate are preserved, interactions that require concentration are stabilized, and energy gradients can be maintained. Each vesicle acts as a miniature laboratory, where molecules can experiment, persist, and evolve. This simple act of boundary formation is profound. Survival begins with separation, the first recognition that persistence requires distinction between inside and outside. These protocells were fragile. A sudden temperature spike, a surge of acidity, or a violent turbulence could rupture a vesicle, destroying its contents. Yet even in their fragility, compartments offered a selective advantage. Molecules that found themselves encapsulated could interact more frequently, catalyzing reactions that promoted replication and energy capture. Networks of molecules, now shielded from the immediate chaos, could establish rudimentary feedback loops. Molecules that stabilized the vesicle or enhanced energy efficiency within it were favored, and through countless cycles of formation, destruction, and reformation, the principles of persistence were quietly encoded in chemical networks. Compartmentalization also amplified the effects of error and variation. Inside a vesicle, an accidental mutation or misfolded RNA strand could produce a reaction that benefits the entire system. Perhaps it stabilizes the vesicle wall, produces a new energy-capturing molecule, or catalyzes the replication of other molecules. The compartment allows these beneficial mistakes to persist long enough to influence the next generation of chemical interactions. In this way, encapsulation is not just protection, it is the enabler of innovation. Life's first steps toward adaptation, cooperation, and complexity could only occur within boundaries. Energy flow within compartments takes on new significance. While external chemical gradients fluctuate unpredictably, encapsulated systems can maintain localized gradients, directing energy more efficiently toward sustaining reactions. Some vesicles may develop primitive mechanisms to harness energy from the environment, such as catalytic surfaces that stabilize energy-rich molecules or chemical networks that shuttle energy between reactions. These early metabolic processes, confined within protective boundaries, allow molecular networks to persist longer and interact more effectively, setting the stage for the emergence of true cellular life. The earliest compartments were likely heterogeneous. Some were formed by lipid assemblies, others by mineral surfaces or naturally occurring cavities in rock. Each type offered different advantages. Lipid vesicles could grow and divide spontaneously under certain conditions, while mineral cavities provided stability against mechanical disruption. The diversity of compartmentalization strategies allowed molecular systems to experiment with various forms of persistence, creating an evolutionary playground long before the emergence of life as we know it. Compartmentalization also introduces the principle of internal organization. Within a vesicle, molecules are no longer randomly distributed, they can concentrate in regions, interact in predictable patterns, and form networks that reinforce themselves. Certain molecules catalyze reactions that produce others, while some provide structural support. Even in the absence of genes, proteins, or true cells, these systems begin to exhibit the hallmarks of life: organization, feedback, and cooperation. Boundaries allow molecular communities to develop strategies for survival that would be impossible in the unbounded chaos of the open ocean. Interactions between compartments themselves may have been an early driver of evolution. Some vesicles could fuse, combining molecular networks and sharing catalytic molecules, while others might fragment, distributing successful networks into new domains. This dynamic interplay introduces another layer of selection. Compartments that harbor efficient, resilient, and adaptable networks are more likely to persist and proliferate. From this simple principle, the foundations of cellular evolution, growth, division, and the inheritance of chemical strategies begin to emerge. Even the smallest compartment represents a triumph against entropy. Molecules trapped within vesicles are shielded from destructive forces, provided with a controlled environment for reactions, and allowed to persist long enough to catalyze new interactions. These boundaries, though ephemeral, teach a fundamental lesson: survival is impossible without protection. Energy capture, replication, and innovation are meaningless if the system cannot maintain itself long enough to benefit from them. Compartmentalization is the first acknowledgement that persistence requires a home, a domain in which the fragile processes of early life can endure. Over time, the principles established by compartmentalization would be refined and expanded. Some vesicles develop mechanisms to regulate internal conditions, such as selective permeability to ions or molecules. Others establish more efficient networks of catalysis, harnessing environmental energy with greater precision. Though primitive, these early strategies lay the groundwork for the first true cells, for LUCA, and for the countless lineages that would emerge in the eons to come. Each step, from lipid vesicle to sophisticated membrane-bound cell, is a direct continuation of the lessons learned in these first fragile compartments: protection, organization, energy management, and persistence. It is striking to consider how radical this development was. Encapsulation does not merely preserve molecules, it transforms them. A network of chemical reactions confined within a boundary behaves fundamentally differently from one in the open ocean. Reactions that are fleeting in bulk water can persist inside vesicles. Networks can cooperate, errors can accumulate as innovations, and energy can be harnessed more efficiently. Compartmentalization turns chemistry into protobiology, creating the conditions for persistence, innovation, and the eventual emergence of life. The emergence of compartments also emphasizes the intimate relationship between life and environment. Boundaries are meaningless without the chaotic, energy-rich surroundings that make persistence valuable. The instability of early Earth, the fluctuating temperature, the chemical gradients, the constant bombardment, created the pressures that made compartmentalization advantageous. Life did not arise in a vacuum, it arose as a response to challenge, as chemistry learned to carve out safe spaces within a violent world. In the grand arc of life's history, compartmentalization represents a turning point. Before membranes existed, molecules could persist only briefly, interactions were chaotic, and the potential for complexity was limited. With boundaries, persistence becomes possible, networks form, and the first strategies for survival, energy capture, and reproduction emerge. These principles, honed over millions of years, are inherited by every living cell today. Every organism, from the simplest bacterium to the most complex human brain, owes its existence to the first fragile compartments that taught chemistry to protect itself. Compartmentalization is more than a structural innovation, it is the first assertion of identity. By establishing an inside and an outside, protocells create a domain where order can persist, interactions can stabilize, and energy can be harnessed. This tiny act of boundary formation is the precursor to individuality, heredity, and ultimately evolution. Without it, life would remain impossible, a fleeting dance of molecules doomed to dissipate. With it, persistence becomes feasible, innovation becomes possible, and the path toward LUCA and all subsequent life is laid. Within the fragile compartments of early protocells, the delicate dance of replication began to evolve. Life had not yet emerged, but matter was learning the first rules of continuity, how to preserve information, maintain organization, and pass it forward. Simple RNA strands, peptides, and lipids were no longer isolated participants in chemical chaos, they were becoming part of integrated systems, networks capable of influencing their own persistence. Replication, once a random and fleeting occurrence, was beginning to refine itself into a process that could sustain and amplify life's earliest principles. At first, replication was crude and error-prone. RNA strands copied themselves imperfectly, sometimes producing sequences that were destroyed immediately, sometimes generating variations that persisted longer. These mistakes, while destructive in isolation, were the drivers of innovation. Some deviations enhanced catalytic efficiency, stabilized compartments, or improved energy utilization. Through countless cycles, molecules that could reproduce more reliably, even imperfectly, became more prevalent. Replication was no longer merely the duplication of molecules, it was a mechanism for selection, adaptation, and eventual complexity. Within the vesicles, the process of replication was shaped by intimate interactions between molecules. Certain RNA strands acted as templates, while others catalyzed reactions that synthesized complementary sequences. Lipids and peptides facilitated stability, maintaining the integrity of the compartment and concentrating reactants. In this confined environment, replication was not a solitary event, it became a networked phenomenon. The survival of one molecule depended on its neighbors, the local chemical environment, and the energy available. Replication evolved as a systemic process, an emergent property of complex chemical networks interacting within protective boundaries. The evolution of replication also reflects a subtle form of cooperation. Molecules that catalyzed the replication of others indirectly increased their own persistence by stabilizing the network. Errors that improved replication efficiency, even modestly, propagated through the system. Beneficial interactions accumulated, creating networks of interdependence that resemble the earliest ecological dynamics at a molecular scale. This is a profound insight. Even before life existed, cooperation, variation, and selection were shaping the trajectory of persistence. Energy remains a central requirement for replication. The synthesis of new molecules consumes chemical energy, and the compartmentalization of early protocells allows energy to be harnessed more effectively. Catalytic molecules help direct energy flows to critical reactions, while gradients within vesicles drive the formation of bonds that would otherwise be improbable. Replication, therefore, is not simply about copying, it is about energy management, timing, and spatial organization. Systems that optimize these factors persist longer, creating feedback loops that refine the process over countless generations. Error, paradoxically, becomes a key component of refinement. Perfect replication would produce homogeneity, a static system incapable of exploring new possibilities. Imperfections introduce variation, and variation allows experimentation. Some errors are catastrophic, destroying strands or destabilizing compartments, but others produce novel sequences capable of new catalytic functions. Over time, these innovations are preserved and integrated into molecular networks. Replication, therefore, is both a conservative and creative force. It maintains continuity while simultaneously generating the raw material for adaptation. Compartmentalization amplifies the impact of replication. Within vesicles, successful molecular networks can persist, replicate, and expand, while destructive events are localized and contained. This isolation enables the accumulation of beneficial configurations, allowing the system to evolve greater complexity. Vesicles that support more efficient replication are more likely to survive, divide, or fuse with others, spreading successful networks. This dynamic introduces a form of natural selection at the level of compartments, long before true cellular life emerges. The interplay between replication and metabolism is also critical. Catalytic networks not only facilitate the synthesis of new molecules but also manage energy flow within the compartment. Replication consumes energy, but it also produces intermediates that stabilize structures, repair damage, and support further reactions. In this way, replication and metabolism co-evolve, reinforcing each other. Systems capable of integrating both processes gain a decisive advantage, persisting longer and creating the first rudimentary feedback loops that characterize living systems. Over countless cycles, replication becomes increasingly refined. Molecular arrangements that reproduce more reliably, manage energy more efficiently, and interact favorably with their compartment and neighbors are favored. This refinement is not conscious, it is the product of persistence under environmental pressures and chemical constraints. Yet the outcome is profound, the emergence of systems capable of continuity, capable of sustaining themselves long enough to accumulate complexity, and capable of laying the foundation for life. It is within these replicated networks that the earliest forms of heredity emerge. Information is encoded in molecular sequences, transmitted across generations, and subject to variation. Beneficial patterns are preserved, while less stable configurations disappear. The protocells are not alive, but they are learning the essential rule of persistence. Survival depends on the faithful propagation of structure, adapted through error and refinement to the challenges of a hostile environment. This principle will be carried forward into every organism descending from LUCA. Replication also introduces a temporal dimension to persistence. Before molecules could reproduce, events were fleeting, disconnected, and ephemeral. With replication, the history of a molecule influences the future. Sequences, folds, and interactions are transmitted across cycles, creating continuity that spans time. In effect, replication allows matter to remember its past configurations, using that memory to influence future survival. This temporal continuity is a hallmark of life, and it begins long before the first cell emerges. The emergence of replication within compartments marks a turning point in the journey from chemistry to biology. Molecules are no longer passive participants in chaos, they are part of networks capable of self-maintenance, self-propagation, and adaptation. These early systems demonstrate the critical principles that define life: energy capture, error-driven innovation, cooperation, and temporal continuity. Replication is not a single reaction but an emergent property of complex interactions, shaped by compartmentalization, catalysis, and environmental pressures. Ultimately, the refinement of replication sets the stage for LUCA. By the time the last Universal Common Ancestor appears, these principles are fully encoded: molecules replicate, networks persist, errors generate variation, energy is harnessed efficiently, and compartments maintain organization. Every cell today is a continuation of this legacy, a testament to the lessons learned in the earliest cycles of molecular replication. The invisible, meticulous refinement of replication is the bridge that connects prebiotic chemistry to the emergence of life itself. Replication teaches a profound lesson: continuity is not guaranteed, it must be earned. The systems that survive are those that manage energy, exploit boundaries, tolerate error, and harness interactions within their network. Fragility and persistence coexist, innovation arises from imperfection, and continuity is built from countless microdecisions at the molecular level. Through these processes, matter transitions from chaos to organization, from randomness to systems capable of enduring across time. In the end, replication is more than duplication, it is the first form of memory, the first mechanism of inheritance, and the engine of evolution long before true life exists. Within the tiny, invisible compartments of early Earth, molecules learn to reproduce, to influence their environment, and to preserve patterns across generations. These lessons, forged in the crucible of chaos, energy, and error, are inherited by LUCA and every living organism today. From these humble beginnings, the story of life, continuity, and persistence unfolds across billions of years, a narrative written in molecules long before there was anyone to witness it. After billions of years of incremental chemical innovation, the molecular chaos of early Earth began to give rise to something extraordinary: LUCA, the last Universal Common Ancestor. LUCA was not the first living thing, nor the first cell, but it was the culmination of countless generations of molecular networks, compartmentalized protocells, and refined replication cycles. In LUCA, the principles that had governed prebiotic chemistry, compartmentalization, energy capture, replication, error-driven innovation, and persistence, coalesced into a system capable of sustaining itself, reproducing with fidelity, and influencing its environment. It was, in essence, the first organism whose descendants would inherit the legacy of all life on Earth. LUCA's emergence was not sudden, it was the product of an unbroken chain of chemical and molecular refinement. For millions of years, RNA strands had folded, replicated, and catalyzed reactions within protective vesicles. Lipid membranes had evolved, forming the first boundaries that separated internal chemistry from external chaos. Early metabolic networks had learned to harness energy gradients from hydrothermal vents, tidal pools, and fluctuating mineral-rich environments. Errors in replication, far from being destructive alone, had generated variation, innovation, and the first primitive feedback loops. LUCA represents the moment when all these principles converged into a coherent, stable system capable of enduring the relentless instability of early Earth. LUCA was microscopic, invisible to the naked eye, yet it carried within it the full blueprint of life's essential principles. It contained the molecular machinery for replication, rudimentary metabolism, and compartmentalization. It could harness energy, repair damage, and tolerate environmental fluctuations. It encoded information, not in a conscious or intentional way, but chemically, in sequences of nucleotides that would propagate through subsequent generations. In this sense, LUCA was both an endpoint of prebiotic chemistry and a beginning, the ancestor of every living organism on the planet today. The environment in which LUCA emerged was harsh, volatile, and unforgiving. Hydrothermal vents erupted unpredictably, tidal pools experienced extreme fluctuations in temperature and salinity, and chemical gradients shifted constantly. Yet LUCA thrived because it embodied resilience. Its compartmentalized structure provided protection, its metabolic networks allowed efficient energy utilization, and its replication systems enabled the faithful transmission of information, with occasional errors fostering further adaptation. LUCA represents life's first mastery of persistence, the first instance where a molecular system could endure, adapt, and propagate despite the relentless challenges of its environment. LUCA's genetic machinery was simple compared to modern organisms, but profoundly sophisticated in its context. RNA, or a derivative thereof, stored information and catalyzed reactions. Ribozymes and simple peptides facilitated key metabolic processes. Lipid membranes maintained boundaries and concentration gradients. This integrated system allowed LUCA to survive, reproduce, and pass on its molecular blueprints. It was not conscious, aware, or purposeful, yet it was alive in the most fundamental sense, a self-sustaining, self-propagating entity capable of influencing its own persistence. Errors, which had driven innovation for millions of years, continued to shape LUCA's evolution. Mutations in nucleotide sequences occasionally led to new catalytic capabilities, improved metabolic efficiency, or enhanced structural stability. Those variations that increased persistence were naturally retained, while detrimental changes were eliminated. This is the principle of natural selection, already encoded at a molecular level. LUCA did not intend to evolve, but its internal mechanisms and interactions with the environment ensured that only the most resilient and adaptable systems would endure. It was the first life form to exist fully within the framework of evolution as we understand it today. Metabolism within LUCA was rudimentary but effective. Chemical gradients from its environment powered the synthesis of energy-rich molecules. Catalytic networks directed energy toward replication, repair, and compartmental maintenance. LUCA had mastered the art of persistence. It could stabilize itself, exploit available resources, and maintain its internal order against the entropic tendencies of the world around it. In doing so, LUCA became the first organism capable of sustaining life long enough to pass on its principles to future generations. LUCA's compartmentalization was equally critical. Its lipid membrane not only separated internal chemistry from the chaos of the external world but also facilitated control over energy fluxes, concentration of molecules, and localized chemical interactions. Within this domain, feedback loops could emerge, catalytic networks could cooperate, and replication could occur reliably. LUCA demonstrates that life survival depends as much on boundaries and organization as on energy capture or replication. Without compartmentalization, persistent life as we know it would have been impossible. One of LUCA's most remarkable legacies is its universality. All known life on Earth descends from this single ancestral node, inheriting its fundamental principles. The genetic code, cellular membranes, metabolic pathways, and replication machinery present in LUCA were transmitted, modified, and refined over billions of years. From bacteria to humans, every organism carries echoes of LUCA's strategies for survival, compartmentalization, replication with variation, energy management, and resilience against chaos. LUCA is not just a historical milestone, it is the chemical and organizational blueprint that underpins all life today. LUCA's existence also underscores the improbability and yet inevitability of life. The precise combination of chemical networks, environmental conditions, and compartmental structures required to produce a persistent, self-propagating system is staggering. Yet given the relentless experimentation of prebiotic chemistry, the accumulation of errors, and the selective pressures of the environment, LUCA emerges not as a miracle but as the natural outcome of persistence, adaptation, and cumulative refinement. Life is improbable, yet inevitable once the conditions allow matter to learn the principles of survival. LUCA also represents the intersection of continuity and innovation. While it preserved the successful strategies of its chemical predecessors, it also introduced subtle variations that would fuel evolutionary diversification. Mutations in genetic sequences, refinements in metabolic pathways, and adjustments in membrane composition created the potential for new lineages to diverge, explore novel ecological niches, and adapt to changing environments. LUCA is simultaneously an end and a beginning, the culmination of prebiotic experimentation and the progenitor of all biological diversity. The philosophical significance of LUCA is profound. It is the bridge between chemistry and life, the moment when persistence, organization, and information coalesce into a system capable of enduring indefinitely through replication and adaptation. It teaches that life is not an external miracle imposed on matter, but a natural consequence of matter's ability to learn, persist, and adapt. LUCA embodies the principles that govern all living systems: continuity through replication, innovation through error, resilience through compartmentalization, and energy management through metabolism. From LUCA, life radiates outward, evolving into the complexity we observe today. Each lineage, each organism, and each cell carries a trace of the strategies first perfected by this ancestral entity. The principles encoded in LUCA are timeless. They are the chemical and organizational foundations that allow life to persist against entropy, adapt to changing environments, and generate diversity. Understanding LUCA is not merely understanding a historical moment, it is understanding the universal rules that govern life itself. In the grand tapestry of Earth's history, LUCA stands as a singular node connecting the chaotic prebiotic past with the vibrant diversity of life that follows. It is a testament to the power of persistence, the importance of boundaries, the inevitability of innovation, and the necessity of energy. From the first RNA strands to the emergence of LUCA, matter has learned to endure, organize, and transmit information across time. LUCA embodies the principles that make life possible and provides the foundation for every living organism that has ever existed or will ever exist. From the moment LUCA existed, life's story began to branch in innumerable directions. LUCA was not a solitary, static entity, it was a nexus, a node from which countless lineages would diverge, each adapting, innovating, and surviving in response to the pressures of a volatile Earth. The diversification of life from this common ancestor marks one of the most profound chapters in the history of the planet, a transition from a single ancestral system to a complex web of organisms interacting, competing, and cooperating in ways that would ultimately give rise to the three domains of life: bacteria, archaea, and eukarya. In the early oceans, environmental heterogeneity drove divergence. Hydrothermal vents provided mineral-rich energy gradients, tidal pools offered fluctuating chemical and thermal conditions, and volcanic islands created pockets of isolation. LUCA's descendants encountered these diverse habitats, each demanding specific adaptations. Small differences in replication fidelity, metabolic pathways, or membrane composition, which might have been inconsequential in a homogeneous environment, suddenly conferred survival advantages. Selection, once operating at the molecular level, began acting on cellular networks, favoring lineages that could exploit niches more efficiently. Over time, this led to the emergence of distinct domains, each with unique strategies for survival, yet all tracing their heritage back to LUCA. Bacteria were among the first lineages to diverge. They thrived in environments where rapid replication and efficient metabolism were paramount. Their relatively simple cellular structures allowed them to exploit a wide range of habitats, from deep-sea vents to shallow coastal pools. Bacteria refined metabolic strategies, developing photosynthesis, nitrogen fixation, and other pathways that would reshape the planet's atmosphere and biosphere. In this lineage, we see the early principles of adaptability, energy management, and compartmentalized replication perfected on a cellular scale. Bacteria demonstrate that persistence is not about complexity, but about efficiency, flexibility, and the ability to endure environmental fluctuations. Meanwhile, archaea diverged along a different path. Many archaeal lineages adapted to extreme environments: boiling hydrothermal vents, hypersaline lakes, acidic springs, and other conditions lethal to most life forms. These extremophiles retained the core principles inherited from LUCA: robust membranes, efficient energy utilization, and adaptive replication, while modifying molecular components to tolerate stress. Archaea reveal life's ability to innovate at a chemical and structural level, demonstrating that survival is possible even in conditions that seem hostile beyond comprehension. Their existence underscores a philosophical truth: life is not constrained by human notions of habitability, it is defined by the capacity to persist under conditions that challenge the very concept of stability. Eukarya, the domain that would eventually give rise to multicellular organisms, animals, plants, and humans, represents a radical innovation in the story of life. Eukaryotic cells evolved larger, more complex structures, including a nucleus to protect genetic material and organelles to compartmentalize energy production and biochemical reactions. This complexity did not arise overnight. It was the result of countless incremental refinements, symbiotic partnerships, and adaptations to energy limitations. Mitochondria, for example, are descendants of ancestral bacteria that were engulfed and retained, providing the cell with unprecedented energy efficiency. The emergence of eukaryotes illustrates how cooperation, compartmentalization, and energy capture, principles perfected in LUCA and prebiotic systems, can combine to produce entirely new levels of biological organization. The divergence into three domains also emphasizes the importance of environmental interaction in shaping evolutionary trajectories. Life did not diversify in isolation, it was constantly responding to chemical, thermal, and ecological pressures. Metabolic innovations allowed some lineages to colonize new habitats, creating feedback loops where environmental modification by one group enabled the survival of others. Photosynthetic bacteria, for example, transformed the atmosphere by producing oxygen, which in turn allowed aerobic metabolism to evolve and ultimately set the stage for complex multicellular life. The diversification of life is thus a story of coevolution. Organisms shape their environment even as the environment shapes them. Errors and variation, once the driving forces at the molecular level, continue to guide diversification. Mutations, gene duplications, and horizontal gene transfer introduced novel capabilities, enabling lineages to exploit new resources or tolerate new stresses. Natural selection, acting on these variations, amplified advantageous traits, gradually producing the immense diversity observed across the three domains. In this way, the same fundamental principles that governed the persistence of molecules in the RNA world now orchestrated the evolution of entire cellular lineages. The process is both continuous and cumulative. Innovations are preserved, refined, and transmitted, creating a cascade of complexity that spans billions of years. Horizontal gene transfer, particularly in bacteria and archaea, highlights another aspect of life's early diversification. Unlike vertical inheritance, where traits pass from parent to offspring, horizontal transfer allows genes, and therefore innovations, to move between unrelated organisms. This accelerates adaptation, spreads metabolic capabilities, and blurs the boundaries between lineages. Early Earth was a crucible of genetic experimentation, and LUCA's descendants exploited every available mechanism to optimize persistence. Diversity, in this context, is not merely the product of isolation and mutation but also of networked interaction, cooperation, and the sharing of innovations across life forms. The emergence of the three domains underscores the duality of life, unity and diversity. All life shares fundamental principles: genetic replication, metabolic networks, compartmentalization, and the management of energy. Yet these same principles give rise to astounding diversity when acted upon by environmental pressures, errors, and innovation. Bacteria, archaea, and eukarya are variations on a common theme, each exploring different solutions to the problem of persistence. This duality reflects the profound continuity of life. Diversity arises not from breaking the rules of survival but from exploring the many possibilities encoded within them. Philosophically, the diversification of life illustrates the interplay between chance and necessity. Life's principles, established in LUCA and prebiotic systems, constrain the possibilities. Replication, energy capture, and compartmentalization are non-negotiable. Yet within these constraints, chance, mutations, environmental fluctuations, and stochastic events, creates novelty, producing the branching tree of life. Diversity is therefore neither random nor predetermined, it is the emergent product of persistence interacting with opportunity over vast spans of time. The story of life's diversification also highlights the invisible scaffolding laid down by LUCA. Every innovation in metabolism, replication, and compartmentalization traces its lineage back to this ancestral system. Each domain represents a unique exploration of the same foundational principles. Bacteria perfected efficiency, archaea mastered resilience, and eukarya discovered new organizational complexity. Yet all retain the legacy of LUCA's strategies for persistence, error-driven innovation, compartmentalization, energy capture, and replication. Without this ancestral foundation, the remarkable diversity of life would have been impossible. In the grand narrative of Earth's history, the divergence from LUCA to the three domains is both a scientific and philosophical milestone. It illustrates how life evolves from a single origin point into a web of interdependent systems, each exploiting niches, energy flows, and opportunities for survival. It shows how persistence, refined over billions of years, can give rise to complexity, adaptability, and ecological interconnectedness. From a solitary ancestral node, life radiates outward, diversifying endlessly, exploring every corner of possibility allowed by the principles encoded in LUCA and its prebiotic predecessors. The three domains remind us that life is both resilient and innovative. It thrives not by adhering to static forms but by continuously exploring, adapting, and exploiting the environment. Diversity is the consequence of persistence and interaction, an ongoing experiment in survival conducted over eons. In every microbe, archaean, or eukaryotic cell, we can trace the legacy of LUCA and the countless molecular experiments that preceded it. Life's diversification is therefore a testament to the enduring principles of chemistry, energy, and persistence that began long before the first organism emerged. The Earth that LUCA and its descendants inherited was far from gentle. It was a world defined by extremes, scalding hydrothermal vents belching mineral-rich fluids into dark oceans, acidic pools that boiled under the relentless sun, glaciers grinding the land in cycles of ice and fire, and volcanic islands erupting unpredictably with molten fury. In this environment, persistence was not guaranteed, survival required constant adaptation. Life, from its earliest cellular forms, was forced to learn resilience, and the pressures of these extremes became the crucible in which biological innovation was forged. Hydrothermal vents, deep in the ocean where sunlight never penetrated, provided a unique combination of heat, minerals, and chemical gradients. For early life, these environments were both a threat and a resource. High temperatures could destabilize membranes, denature molecules, and destroy fragile chemical networks. Yet the same conditions provided abundant energy gradients and reduced compounds that could fuel metabolism. LUCA's descendants, and the lineages that followed, evolved mechanisms to exploit these extremes: membranes capable of resisting thermal disruption, enzymes adapted to function at high temperatures, and metabolic pathways optimized for energy extraction from inorganic compounds. Extremes, in this sense, became opportunities for life to innovate. Surface environments presented different challenges. Tidal pools, with fluctuating salinity, temperature, and pH, forced early organisms to tolerate variability. Organisms that could withstand desiccation, ionic stress, or sudden chemical shifts had a selective advantage. These pressures encouraged the evolution of more robust membranes, stress-tolerant enzymes, and repair mechanisms that maintained cellular integrity under fluctuating conditions. Life was learning to endure instability, to turn environmental volatility from a source of destruction into a driver of adaptation. The chemical composition of early Earth also played a critical role. The abundance of minerals such as iron, sulfur, and magnesium created natural catalysts that facilitated energy-capturing reactions. Life did not invent chemistry, it co-opted it. Microbes evolved to exploit these minerals, using them to drive metabolic reactions that would have been impossible otherwise. The interplay between environment and biology created feedback loops. The chemical landscape shaped the evolution of metabolism, which in turn altered the local environment, producing new selective pressures. In this way, Earth itself became a partner in life's innovation. Volcanic activity and geothermal gradients provided pulses of energy that early organisms could exploit. Sudden surges of heat or reactive chemicals could destroy unstable networks but also catalyze novel reactions, producing compounds that were otherwise inaccessible. Life learned to harness these energy bursts, stabilizing reactions within compartments and redirecting energy toward replication and persistence. Extremes, whether destructive or energetic, were inseparable from opportunity. The very forces that threatened survival also created the conditions for innovation and adaptation. Oxygen, initially toxic to most early life, became a transformative environmental pressure. As photosynthetic bacteria evolved and released oxygen into the atmosphere, LUCA's descendants were faced with a deadly challenge. Organisms either developed mechanisms to tolerate oxygen, exploited it for energy, or sought refuges in anaerobic environments. This selective pressure drove the evolution of antioxidant enzymes, new metabolic pathways, and eventually complex aerobic respiration. Once again, the environment forced innovation. Life did not passively exist in the face of challenges, it evolved in direct response to them. Earth's extremes also promoted specialization and diversification. Organisms adapted to deep-sea vents were different from those in tidal pools, hypersaline lakes, or acidic springs. Environmental pressures drove lineages into distinct ecological niches, fostering the development of unique metabolic strategies, structural adaptations, and reproductive mechanisms. Life became modular and flexible, capable of exploring multiple survival strategies simultaneously. Extremes, by imposing boundaries and constraints, created a landscape of evolutionary opportunity. Temperature fluctuations, from searing heat to chilling cold, further shaped life. Molecules and membranes were forced to tolerate or exploit these changes. Proteins evolved to remain stable across temperature ranges, lipid membranes adjusted composition to maintain fluidity, and metabolic networks adapted to variable rates of energy flow. Life's molecular machinery was tested continuously, refined by cycles of stress and recovery. In doing so, organisms learned to persist not only in static conditions but in the dynamic, unpredictable world that Earth presented. Environmental pressures also fostered cooperation. Within extreme habitats, organisms that could interact synergistically often had higher survival rates. Metabolic byproducts from one species could fuel the growth of another, forming early microbial communities. Symbiotic relationships emerged, some transient, others permanent, creating networks of interdependence that enhanced collective persistence. Extremes, by challenging survival, encouraged not only innovation within individual organisms but collaboration across lineages. Cooperation became a strategy as vital as adaptation or replication. Earth's chemical instability created selective pressures that shaped the very building blocks of life. Extremes favored molecules and structures that were resilient, RNA that folded reliably, lipids that maintained membrane integrity, proteins that resisted denaturation. Molecular selection, operating within environmental constraints, ensured that only the most robust configurations persisted. Life's earliest innovations were therefore directly sculpted by the planet's volatility. Without extremes, many of the mechanisms that define life today, repair, stress tolerance, energy efficiency, might never have arisen. Even stochastic events, such as meteorite impacts or volcanic eruptions, played a role. Sudden, catastrophic disturbances eliminated unstable organisms, opening ecological niches for survivors. Life evolved to exploit these opportunities, dispersing into new habitats, refining stress responses, and diversifying into new forms. Extremes created both filters and opportunities. They eliminated the weak and rewarded those capable of exploiting instability. Evolution became a dialogue with the environment, where survival depended on responsiveness, resilience, and adaptability. Life's responses to extremes also set the stage for later ecological interactions. By colonizing niches ranging from boiling springs to icy glaciers, organisms began to shape local chemical and physical conditions. They modified energy flows, produced byproducts that influenced other species, and created microenvironments that further selected for adaptation. Extremes were not merely obstacles, they became a canvas on which life painted its first ecological strategies. Survival required both adaptation to and manipulation of the environment, laying the foundations for ecosystems that would eventually sustain complex multicellular life. Reflecting on the role of extremes in life's early history reveals a profound lesson: challenge is a catalyst for innovation. Persistence is not merely the endurance of favorable conditions but the capacity to thrive amid volatility. The earliest organisms, forged by hydrothermal vents, tidal pools, and chemical fluctuations, learned to harness instability as a tool. Energy capture, compartmentalization, replication, and error-driven adaptation were all refined under environmental pressure. Extremes were not enemies of life, they were its instructors. The shaping influence of extremes is evident in the diversity of modern life. Extremophiles, thriving in conditions once thought uninhabitable, are direct descendants of these early experiments in persistence. They embody the lessons learned over billions of years: resilience, adaptability, cooperation, and innovation under stress. Life's very existence on Earth is a testament to the power of environmental pressure to forge complexity, diversity, and stability from chaos. Ultimately, the story of life under extremes is a story of emergence. LUCA's descendants, molded by environmental pressures, adapted, diversified, and innovated. Through repeated cycles of challenge and survival, life transformed from fragile protocells into the robust, dynamic, and interconnected web of organisms we observe today. Extremes shaped metabolism, replication, compartmentalization, and cooperation, embedding resilience and adaptability into the very fabric of biology. Without these harsh lessons, life might have remained ephemeral, a fleeting chemical curiosity rather than the persistent, evolving phenomenon that fills every corner of the Earth. In the crucible of extreme environments, life learned its most essential lesson: persistence is not passive. It requires energy, structure, adaptation, and responsiveness. Earth's extremes did not simply challenge life, they taught it how to endure, innovate, and flourish. The legacy of this shaping continues in every organism, from the simplest microbe in a boiling spring to the most complex mammal navigating a temperate forest. Life, in every form, carries the memory of Earth's harshest conditions and the strategies that allowed survival against impossible odds. In the unfolding saga of life, the emergence of molecular memory marked a pivotal moment, one that transformed ephemeral chemical interactions into enduring legacies. RNA, which had dominated prebiotic systems and early protocells, was both information carrier and catalyst, a dual role that allowed chemical networks to persist and adapt. Yet RNA was fragile, prone to hydrolysis and errors, and its capacity for long-term continuity was limited. The transition to DNA, a more stable repository of genetic information, represents one of life's most profound innovations. Through this shift, the principles of replication, error correction, and inheritance became encoded with unprecedented fidelity, allowing life to carry memory across generations, to refine itself cumulatively, and to explore the vast potential of complexity. RNA's role in molecular memory was extraordinary. In addition to serving as a template for replication, RNA molecules catalyzed chemical reactions, folding into three-dimensional structures capable of accelerating specific transformations. This dual functionality allowed early life to encode information and act upon it simultaneously. RNA strands could propagate sequences that worked, catalyze reactions that generated energy or building blocks, and influence their own persistence. Within compartments, networks of RNA catalysis became self-reinforcing. Successful sequences promoted replication, energy capture, and compartment stability, while less effective sequences faded into chemical oblivion. In this way, RNA established the first primitive memory systems, linking the past to the future through chemical continuity. Yet RNA's fragility posed limits. Hydrolysis, thermal fluctuations, and replication errors meant that information could be lost as easily as it was transmitted. Short-lived strands and high mutation rates constrained the accumulation of complex networks. Life needed a more reliable medium to preserve the lessons learned over millions of years of experimentation. DNA, with its double-stranded structure and chemical stability, provided that medium. By separating the roles of catalysis and information storage, DNA allowed RNA to continue its catalytic functions while preserving sequences with far greater fidelity. This division of labor between RNA and DNA represents one of life's earliest optimizations, a leap from ephemeral chemical persistence to enduring hereditary continuity. The transition to DNA-based molecular memory also allowed the gradual expansion of complexity. With information preserved in a stable format, organisms could accumulate innovations over countless generations. Mutations and recombinations in DNA introduced variation, while the molecule's stability ensured that advantageous configurations were retained. Feedback loops between replication fidelity, mutation, and selection drove cumulative evolution, allowing lineages to explore adaptive landscapes that were inaccessible to the fragile RNA-only systems. Life, for the first time, could remember and refine strategies, preserving the successes of past generations while experimenting with new possibilities. Molecular memory also enabled the evolution of regulation. RNA and DNA sequences could encode instructions for producing molecules at specific times or under specific conditions, coordinating metabolic reactions and replication processes. Within compartments, this regulation allowed systems to respond dynamically to environmental fluctuations. Feedback mechanisms emerged. If energy resources dwindled, replication slowed. If stress increased, repair mechanisms intensified. The capacity to remember environmental cues and adjust internal processes marked a profound step towards cellular intelligence, where life was no longer a passive chemical reaction but an adaptive system capable of responding to challenges over time. The chemical stability of DNA also facilitated the emergence of the genetic code, a universal system that translates nucleotide sequences into functional proteins. Proteins, in turn, became specialized catalysts and structural elements, expanding the range of reactions and interactions that cells could perform. This codified relationship between sequence and function created a more robust form of memory. Information encoded in DNA could reliably produce molecules that influenced persistence, metabolism, and replication. Life had evolved from transient chemical networks to a system capable of storing lessons, directing activity, and transmitting these capabilities across eons. RNA retained its catalytic importance, serving as a mediator between DNA and protein synthesis, particularly in the form of ribozymes and messenger RNAs. This interplay created a dynamic memory system. DNA preserved information, RNA interpreted it, and proteins executed the functions necessary for survival. The separation of these roles optimized persistence, allowing errors in one domain to be tolerated while innovations could be preserved and propagated. Molecular memory became multi-layered, with redundancy, feedback, and adaptability built into the very fabric of cellular systems. Error correction mechanisms further enhanced the fidelity of molecular memory. DNA polymerases and repair enzymes evolved to identify mismatched bases, excise them, and replace them with the correct sequence. These mechanisms reduced the destructive consequences of errors, allowing beneficial mutations to accumulate without destabilizing the entire system. Life had learned not only to remember but to protect its memory, ensuring that the innovations accumulated over billions of years of molecular experimentation were preserved for future generations. Compartmentalization reinforced molecular memory by isolating systems and concentrating molecules, facilitating efficient replication and energy utilization. Membranes provided not only physical protection but also the conditions necessary for feedback loops to operate reliably. Within these microenvironments, information could be transmitted, chemical networks stabilized, and evolutionary experimentation preserved. Memory, both chemical and informational, depended on boundaries. Without compartmentalization, the fidelity and continuity of RNA, DNA, and protein interactions would have been impossible. Environmental pressures continued to shape molecular memory. Extremes in temperature, pH, salinity, and chemical composition acted as selective filters, favoring sequences that could withstand stress. Innovations in DNA and RNA sequences that enhance structural stability, metabolic efficiency, or replication fidelity were preserved and propagated. Molecular memory, therefore, is not only the product of intrinsic chemical properties but also of interactions with a hostile, dynamic environment. Life's ability to endure, refine, and adapt is inseparable from the selective pressures imposed by Earth itself. The emergence of DNA-based memory also set the stage for the evolution of complexity at higher scales. Multicellularity, differentiation, and eventually the development of nervous systems rely on the precise storage and transmission of information. The strategies first perfected in LUCA, compartmentalization, energy capture, replication, and error-driven adaptation, are encoded in DNA, enabling cells to communicate, coordinate, and respond dynamically to environmental and internal cues. Molecular memory thus underpins the vast tapestry of biological complexity that extends from single-celled organisms to the human brain. Philosophically, molecular memory represents life's first assertion of continuity through time. It allows life to learn from the past, adapt in the present, and prepare for the future. RNA and DNA are not mere molecules, they are repositories of experience, storing the outcomes of billions of years of chemical experimentation and environmental interaction. Every replication, every mutation, every repair event is a conversation between past and present, a negotiation between stability and innovation. Memory is the mechanism by which life preserves the lessons that ensure persistence against entropy. In this sense, life becomes a story written in molecules. RNA catalyzes, DNA encodes, proteins execute, and compartments orchestrate the interactions. Information is preserved, innovations accumulate, and environmental feedback shapes outcomes. Molecular memory transforms ephemeral chemical events into enduring strategies, allowing life to explore complexity and diversity while maintaining continuity. Without this memory, the innovations of prebiotic chemistry, the resilience of LUCA, and the adaptations forged in extreme environments would be lost at every generation, and life as we know it could not exist. Molecular memory also underscores the profound improbability and inevitability of life. The chemical systems that gave rise to RNA, DNA, and proteins required precise conditions, repeated cycles of experimentation, and countless errors. Yet the principles that govern molecular memory, replication, compartmentalization, energy capture, and error-driven refinement, make the persistence of life not a miracle but a consequence of these cumulative processes. Life's continuity is secured by its capacity to remember, adapt, and innovate, linking the earliest chemical networks to the complex organisms that inhabit the Earth today. Ultimately, molecular memory represents the bridge from chemistry to biology. RNA and DNA are the vessels through which matter learns, adapts, and preserves knowledge across time. They enable the accumulation of innovations, the refinement of survival strategies, and the exploration of complexity. Life, in preserving and acting upon this memory, transcends mere chemical existence. Memory allows persistence, continuity, and evolution, ensuring that the lessons of prebiotic chemistry, LUCA, and extreme environments are carried forward into every living organism, shaping the trajectory of life on Earth for billions of years to come. In the ancient oceans and primordial pools where life first emerged, survival was not solely the product of individual persistence. The pressures of a volatile environment, combined with the limitations of molecular replication and energy capture, created a context in which cooperation and symbiosis became powerful strategies for persistence. Life learned, in subtle chemical and cellular ways, that working together could amplify survival, expand ecological reach, and accelerate innovation. Symbiosis, at its core, is the recognition that boundaries between organisms are porous when persistence is mutually advantageous. The earliest forms of cooperation were chemical in nature. Molecules within compartments often catalyzed reactions that benefited their neighbors. A ribozyme might generate a metabolite that stabilizes other molecules or promotes replication elsewhere in the vesicle. Compartments that contained networks with mutually supportive interactions were more likely to persist through environmental fluctuations, while those lacking cooperation were destroyed. At this microscopic scale, symbiosis is not conscious or intentional, it is an emergent property of chemical systems interacting within shared boundaries. The principle, however, would reverberate throughout life's history, collaboration enhances persistence. As cells evolved, these early cooperative interactions became more complex. Microbes in hydrothermal vents or tidal pools could interact metabolically, exchanging chemical byproducts that one organism could exploit while detoxifying the environment for another. For instance, one species might produce a compound that serves as a nutrient for another, while the second species detoxifies a harmful byproduct in return. Such mutually beneficial arrangements created localized networks of persistence, microecosystems in which life could sustain itself more effectively than in isolation. Cooperation allowed life to occupy niches that would otherwise have been inaccessible, expanding the ecological landscape for diversification. Endosymbiosis represents one of the most profound leaps in the history of cooperation. Eukaryotic cells, through the engulfment of bacterial cells that became mitochondria and chloroplasts, internalized what were once independent organisms. This partnership allowed eukaryotes to harness energy far more efficiently than any single cell could alone. Mitochondria provided abundant ATP, fueling the development of larger, more complex cells capable of new metabolic, structural, and reproductive strategies. Chloroplasts, descendants of photosynthetic bacteria, enabled the capture of solar energy and the transformation of ecosystems through oxygenic photosynthesis. Symbiosis was no longer a peripheral strategy, it became a central mechanism for innovation, complexity, and ecological dominance. Cooperation extends beyond metabolic exchange. Cells developed signaling mechanisms to coordinate behavior within colonies, biofilms, and eventually multicellular organisms. Communication allowed groups of cells to regulate replication, respond collectively to environmental changes, and optimize resource utilization. Feedback loops emerged, where signals influenced behavior, behavior influenced survival, and survival reinforced cooperative strategies. Life learned that persistence was not just about individual optimization but about collective strategy. In essence, cooperation became encoded into the very processes of replication, metabolism, and adaptation. Symbiotic relationships are also a vehicle for accelerated evolution. By sharing resources, genes, or metabolic functions, organisms can explore adaptive possibilities that would be inaccessible individually. Horizontal gene transfer, common in bacteria and archaea, is a prime example. Genes that confer new metabolic capabilities, stress tolerance, or replication advantages can move between species, spreading innovations rapidly across populations. Cooperation and symbiosis thus function as multipliers for persistence and innovation, enabling life to experiment and adapt on a scale that would be impossible for isolated organisms. Environmental pressures further amplified the value of cooperation. Extreme conditions, high temperatures, fluctuating pH, chemical toxicity, and resource scarcity, created selective advantages for organisms that could mitigate challenges collectively. Communities of microbes could stabilize local environments, buffer chemical fluctuations, and share metabolic burdens, creating resilience that surpassed what individual cells could achieve. Life in these extreme conditions illustrates that cooperation is not a luxury, it is often a requirement for survival in hostile environments. Cooperation also fosters complexity. By integrating metabolic networks, signaling pathways, and compartmental strategies, organisms can develop hierarchical organization. Multicellularity, a later outcome of these cooperative principles, depends on the ability of cells to specialize, communicate, and persist collectively. Each cell sacrifices some independence for the benefit of the larger system, creating emergent properties such as tissue formation, energy distribution, and developmental programs. In this sense, symbiosis and cooperation are not just strategies for persistence, they are drivers of biological innovation and structural sophistication. Even at the molecular level, cooperation continues to shape persistence. Proteins form complexes, RNA molecules assist in multiple reactions, and metabolic pathways are intricately interconnected. The stability of these networks often relies on synergistic interactions. One molecule alone might be ineffective, but together, they sustain reactions, amplify energy capture, and stabilize compartments. Life demonstrates repeatedly that collective function can exceed the sum of individual capacities, that interdependence enhances survival. Symbiosis is not without tension. Relationships can be exploited, resources can be unevenly distributed, and environmental pressures can shift balances of benefit. Yet these dynamics drive further innovation. Cells develop regulatory mechanisms to maintain cooperation, signaling to coordinate interactions, and structural modifications to reinforce beneficial partnerships. Conflict and resolution within symbiotic systems mirror evolutionary pressures at the molecular level, reinforcing resilience and adaptability. Life, therefore, thrives on the delicate balance between cooperation and competition, between shared benefit and individual persistence. The evolutionary significance of cooperation is immense. It enables rapid exploration of ecological niches, efficient energy utilization, and resilience under fluctuating conditions. Symbiosis fosters diversification by allowing organisms to combine capabilities, innovate collaboratively, and stabilize otherwise fragile networks. In every lineage, from bacteria to eukaryotes, the capacity to cooperate has amplified survival prospects, driven metabolic and structural innovation, and laid the foundation for complex ecosystems. Philosophically, symbiosis reveals that life's persistence is not purely individualistic. Survival is a web of interdependence, an intricate interplay of cooperation, competition, and adaptation. Even the earliest molecular networks hinted at this truth. By supporting each other, molecules increased the likelihood of persistence. Life, at every scale, reflects this principle. From microscopic ribozymes to sprawling multicellular organisms, the capacity to cooperate transforms vulnerability into resilience and chaos into opportunity. The lesson is clear: persistence is a collective endeavor. LUCA's descendants learned that boundaries are not absolute and that collaboration can extend survival, enhance innovation, and accelerate adaptation. Symbiosis and cooperation are not anomalies of evolution, they are central strategies encoded in the fabric of life, emerging first at molecular and cellular levels and culminating in the rich tapestry of ecosystems, multicellularity, and complex behavior. From chemical networks to modern ecosystems, cooperation has remained a constant theme. Life continues to demonstrate that survival and innovation are amplified when entities work together, share resources, and stabilize each other against environmental fluctuations. Symbiosis is a bridge, from the isolated experiments of early molecules to the collective persistence of multicellular organisms and the interdependent networks of life that define our planet today. Ultimately, cooperation and symbiosis teach a profound lesson about life strategy for persistence. Survival is rarely a solo endeavor. It is a networked process, a continual negotiation between independence and interdependence, vulnerability and resilience. The principles first explored in molecular networks, reinforced in early compartments, and refined in LUCA's descendants are visible today in every thriving ecosystem. Life persists not merely through the strength of individual parts, but through the collective intelligence and interdependence encoded into its very fabric. From the very origins of life, energy has been both the constraint and the enabler of persistence. In the chaotic chemical landscapes of early Earth, energy was abundant yet fleeting, available in gradients, heat flows, and chemical potentials, but capturing it reliably was another matter entirely. Life, from its first fragile compartments to LUCA and beyond, learned to transform energy into a currency of survival, establishing metabolic networks that could store, channel, and deploy this power efficiently. Metabolism is life's negotiation with the environment, its mechanism for converting chance availability into predictable sustenance, and its strategies for energy efficiency reveal the deep intelligence embedded in biology's molecular architecture. At the earliest stage, protocells relied on simple redox reactions catalyzed by minerals or RNA molecules. Chemical gradients at hydrothermal vents provided electrons that could be harnessed to drive the formation of energy-rich molecules. Within compartments, these reactions were concentrated, stabilized, and coupled to the synthesis of molecules necessary for persistence. Energy was fleeting, so every molecular interaction had to be efficient, every catalytic cycle optimized. Systems that wasted energy dissipated before replication or repair could occur, while those that captured even a fraction more could persist, replicate, and propagate their chemical strategies. This is the foundation of life's first energy economy, survival dictated by energy efficiency. As replication became refined, so too did metabolic networks. Simple molecules could no longer sustain complex replication or repair indefinitely. Catalytic molecules evolved to facilitate multi-step reactions, creating chains of energy transfer that amplified the utility of available resources. Early metabolism was not merely about survival, it was about optimization. Molecules that could extract more energy from environmental inputs, stabilize intermediates, or redirect reactions toward replication were naturally selected. Life, in this sense, is an energy economist, constantly evaluating costs, maximizing returns, and refining transactions at the molecular level. Compartmentalization amplified energy efficiency. By isolating reactions, protocells prevented the dissipation of intermediates, concentrated substrates, and maintained gradients that could be exploited to drive energetically unfavorable reactions. Lipid membranes acted as insulators, creating microenvironments in which chemical work could be performed predictably. Within these bounded spaces, energy could be stored temporarily, directed toward replication or repair, and conserved for times of scarcity. Compartmentalization and metabolism co-evolved, boundaries enabled energy management, and energy management reinforced the utility of boundaries. The emergence of LUCA brought new sophistication to energy economies. LUCA's metabolic networks were more integrated and capable than those of earlier protocells. Membranes maintained proton gradients that powered ATP synthesis, chemical reactions were organized into cycles, and molecular interactions were orchestrated to minimize energy loss. Replication, repair, and basic maintenance were no longer haphazard, they were fueled by controlled energy flows. LUCA's descendants inherited not only its structural and genetic legacy but also its strategies for energy capture and management. The efficient use of energy became a cornerstone of life's persistence and diversification. Energy efficiency also drove innovation. Organisms that could extract energy from previously inaccessible sources gained a competitive advantage. The evolution of photosynthesis, for instance, transformed life's relationship with sunlight. Organisms that could convert solar energy into chemical energy opened new ecological niches, altered planetary chemistry, and created opportunities for aerobic metabolism. Similarly, chemolithotrophs exploited inorganic compounds such as hydrogen, sulfur, or iron, converting them into energy in environments hostile to other life forms. Every innovation in energy capture was a new solution to the universal problem of survival, making the most of what the environment provides. Errors and mutations in metabolic pathways were not mere accidents, they were opportunities for optimization. Slight modifications in enzymes could improve catalytic efficiency, alter substrate specificity, or stabilize reactions under extreme conditions. Over generations, these small improvements accumulated, creating energy systems that were not only more efficient but more robust. Life, shaped by billions of years of trial and error, learns to economize energy with remarkable precision. In every cell today, from the simplest bacterium to complex eukaryotes, these principles are evident: energy flows are regulated, conserved, and deployed strategically to sustain persistence and promote replication. Symbiosis and cooperation also enhance energy economies. In microbial communities, waste products from one organism often serve as energy sources for another. Metabolic interdependence allows collective optimization. One species may focus on energy-intensive processes, while another efficiently exploits the byproducts. This dynamic creates ecosystems in which energy is cycled efficiently, losses minimized, and survival maximized. Life, at all scales, turns cooperation into an energy-saving strategy, demonstrating that efficiency is not solely molecular but ecological. Extreme environments further refine energy management. Hydrothermal vents, acidic pools, and hypersaline lakes present fluctuating, often hostile, conditions that challenge energy capture. Life adapts by evolving specialized enzymes, flexible membranes, and alternative metabolic pathways to extract energy under these stresses. Extremophiles exemplify how energy efficiency is intertwined with survival strategy. Persistence in harsh environments depends on precise exploitation of available energy and the minimization of wastage. Energy, in these contexts, is both a limiting factor and a driver of innovation. The efficiency of energy capture is also linked to the evolution of complexity. Multicellular organisms, for example, require sophisticated energy distribution networks to maintain homeostasis, coordinate development, and sustain reproduction. Mitochondria, descendants of ancestral bacteria, are a prime example. By internalizing these energy-producing symbionts, eukaryotic cells achieve levels of metabolic efficiency that support larger size, complex organization, and more intricate behaviors. Energy economy, therefore, is not just a molecular necessity, it is a prerequisite for complexity, specialization, and adaptive diversification. Metabolic networks also provide feedback mechanisms that enhance persistence. Energy deficits trigger stress responses, slowing replication or activating repair systems. Surpluses allow accelerated growth, reproduction, or adaptation. These feedback loops, present even in simple organisms, demonstrate that life is not a passive chemical system but a dynamic regulator of energy flows. Metabolism is a form of temporal intelligence. It senses, adjusts, and optimizes, linking immediate environmental conditions with the long-term persistence of the organism. Philosophically, life's energy economy reveals a profound lesson: survival is contingent upon the careful management of limited resources. Life does not merely endure, it transforms energy from chaos into structured work, converting fleeting gradients into stable persistence. From the first redox reactions in primordial pools to the ATP-driven complexity of modern cells, life's ability to economize energy is central to its resilience, adaptability, and creativity. Without efficient energy capture and deployment, replication, repair, and innovation would falter, and persistence would be impossible. Ultimately, energy economies are the invisible architecture of life. They link molecular replication, compartmentalization, error-driven innovation, and symbiosis into an integrated strategy for survival. Life has evolved not only to survive but to do so efficiently, maximizing the utility of every photon, electron, and chemical gradient. From the earliest molecules in chaotic pools to complex eukaryotic organisms orchestrating elaborate metabolic networks, life's story is inseparable from its mastery of energy. Persistence, in every form, is the product of energy management refined across billions of years. In every cell, in every ecosystem, and in every evolutionary innovation, energy is the currency that enables persistence, complexity, and adaptation. The earliest organisms learned that survival was not simply about existence, it was about converting available energy into continuity. Life's economy of energy is both a strategy and a constraint, a driver of adaptation, and a lens through which the entire history of biology can be understood. Mastery of energy underpins every leap from chemical networks to LUCA, from single-celled lineages to multicellular organisms, and from simple persistence to ecological dominance. Life, in all its forms, is a testament to the relentless optimization of energy in the service of survival. Chance has always been a subtle yet profound architect of life's trajectory. From the earliest molecular networks to the complex ecosystems of today, random events, mutations, chemical fluctuations, environmental perturbations, have shaped the path of persistence in ways both unpredictable and transformative. Mutation, once a microscopic error in replication, became the engine of variation, creating new possibilities from the constraints of existing systems. Life, in learning to exploit chance, turned randomness into opportunity, building diversity, adaptability, and innovation from the unpredictable forces of the universe. At the molecular level, chance manifests as replication errors. RNA and DNA, the carriers of molecular memory, are not perfect, every copying event carries a small probability of mistake. In early life, these errors could be catastrophic, destroying sequences or destabilizing compartments. Yet some mistakes produced sequences with enhanced catalytic abilities, more stable folding, or better energy utilization. These beneficial variations were preserved, while detrimental ones were eliminated. Mutation is not intentional, nor guided by purpose, but the persistence of favorable outcomes demonstrates how chance interacts with selection to drive innovation. Environmental fluctuations amplify the role of chance. Changes in temperature, pH, salinity, or chemical availability can alter which variations are advantageous at a given moment. A mutation that confers heat tolerance may be useless in a cooler pool, while a change that improves energy extraction becomes critical when resources are scarce. Life's trajectory is therefore a negotiation between internal variation and external contingency. Evolution is both constrained and liberated. The principles of replication, metabolism, and compartmentalization guide survival, but chance determines which innovations will persist and flourish. Horizontal gene transfer exemplifies the interplay between chance and opportunity. Genes move across lineages unpredictably, introducing new capabilities into organisms that did not evolve them directly. Metabolic innovations, stress tolerance mechanisms, or structural proteins can spread rapidly, altering the evolutionary potential of entire populations. Chance creates access to novel solutions, while selection determines whether these solutions endure. Life thrives in this probabilistic landscape, exploiting stochastic events to expand possibilities and accelerate adaptation.